Good afternoon, everyone. Welcome to the sixth science meeting of PRERNA. And this year, we are having the science meeting on pediatric tuberculosis, a health system perspective. We have all our panelists join here. Our teams are sitting at respective sides. Welcome to all the audience who have joined in the webinar as well. So we start the meeting. Let me invite Dr. Sanjay Juvekar, PI of PRERNA Group, for the welcome remarks.
Thank you so much, Swati, and welcome everybody. Welcome to this platform for research excellence related to national aims, as it is acronymed as PRERNA meeting organized by the three partners of PRERNA on pediatric tuberculosis, a health system perspective. Can we go to the next slide, Rituja? Okay, now PRERNA has been working since, I think, 2012, almost 10 years we've been working together. It is a consortium of about 3 institutions, the CHRD SARS in Delhi, CMC Vellore, and KM Hospital Research Center in Pune. And what does this PRERNA group or the consortium do? It convinces ideas, converted into research proposals that acquired global national funding. It develops manuscripts that are published in peer-reviewed journals. We train science meetings around maternal and child health issues. And we do conduct workshops, training to its vibrant young investigators. These young investigators are the young scientists based at these 3 sites. They are from various fields, from medical background to social science background. From within medical, it is from community medicine to gynecology to pediatrics and various backgrounds. And ultimate aim of PRERNA has been to create leaders who will lead research in different domains around maternal and child health in India. And that's what we've been trying to do, and we've been successfully doing. Can we go to the next slide, Rituja? So I'm just giving a few photographs you could see here. These are the young scientists who graduated as the PRERNA young investigators in the past 10 years. And you would see that many of these are placed at different national level, global level institutions in the country. And all these are now emerging as leaders in research in India. We are very proud of these young investigators who have graduated from PRERNA. And currently we have yet another set of 8 investigators, again at these 3 sites, who would be graduating very soon before we complete 10 years of working as PRERNA partners. So these 10 years have been funded by the Bill and Melinda Gates Foundation. And we hope that the PRERNA platform continues to strive and develop more and more young investigators to become investigators in their respective fields or their respective domain of interest. Can we go to the next slide? This year, as we've been doing science meetings all these years, but this time we thought, let's look at the pediatric TB, a matter of concern for everyone in this country, in many of the LICs as well as LMICs. And even for that matter, HICs, high-income countries, they are not also spared of TB. So it is a concern, especially the pediatric TB is a concern, especially when we know that more than 70% of children with TB across the world are missed being notified. What does that mean? We do not know how many of these, who are these. If these are more than 70% who are missed being notified, which is very likely to say that many of these are deprived of getting the due treatment for tuberculosis as per the WHO norms. Near about 16% of the children with TB die. And this is not a small number. Worldwide, it is 1.4 million. It's a big number. If you talk about our own country, 31% of the global pediatric TB cases are from our own country alone. So it tells us the magnitude of this pediatric TB, a menace for the country. About 5.56% of notified TB cases in India are pediatric TB, which is less than 14 years of age, which means we are a long way to go. If we notify all the pediatric TB cases, I think the numbers will be much higher. I think that is why we thought, as PRERNA Group, should we try and understand pediatric TB as a whole? Can we go to the next slide? So what are we going to do in these 2 days? Today, the chair for this will be Dr. Varinder Singh and co-chaired by Dr. Rakesh. Dr. Varinder Singh will also begin to tell us his journey as a clinician from diagnosis to the completing of treatment by children for tuberculosis. And then Dr. PRERNA will take us through the diagnostic gamut in pediatric TB that is available and used in the country. And then Dr. Aishwarya will tell us a bit about the vaccines in tuberculosis. And the last presentation of the day will be by Dr. Christian, who will talk about the TB infection and importance of TB preventive therapy. Tomorrow, we are going to hear another 3 great presentations. First one would be by Dr. Sanjay Mattu, who will tell us about the perspective of National TB Elimination Program with respect to pediatric TB. And then Dr. Vijayashree will tell UHC initiatives and implications on TB care services delivery. And Dr. Rakesh will tell us about engaging private sector, along with Dr. Pranati, who is also going to tell us about on the engaging private sector effectively for pediatric TB. We know that in our country, more than 60% of the people, the first approach is always a private sector. If that big numbers are going to the private sector, we cannot neglect or ignore the private sectors involved in TB control. And that's why it is important. And lastly, Dr. Devadasan is going to tell us why do we need systems thinking to tackle the problem of pediatric TB? In spite of having a large private sector, we cannot deny the fact that in countries like India and many other LMICs or LICs also, health system perspective is the way forward for getting rid of tuberculosis. So that's what we are going to talk in today and tomorrow. And over to you, Swati.
Thank you, sir. So we'll start the sessions for the day. Let me begin by introducing the chairpersons for today. We have 2 eminent people as our chairpersons for the sessions today. Dr. Varinder Singh is a professor of pediatrics at Lady Hardinge Medical College and associated Kalavati Saran Children's Hospital in Delhi. And he's also the director professor of pediatrics, cum lead of the National Center of Excellence for Pediatric TB under NTEP. He is also the lead of National Technical Expert Group for Pediatric TB for NTEP. He has contributed to the Government of India and the Indian Academy of Pediatrics guidelines on asthma, TB and respiratory infections, and COVID in children. He has authored several papers, chapters, and books. His areas of work include pulmonology, intervention bronchology, TB, rational antimicrobial therapy, research methodology, and innovations in health. Welcome, Professor Varinder Singh. Our co-chair for the day is Dr. Rakesh PS, who's a medical doctor with post-graduation in public health. He has been working for TB elimination in various roles for the past 10 years. Currently, he is the Senior Technical Advisor at the Union Southeast Asia Office. He was a WHO consultant to National TB Elimination Program, and his key contributions during that period included framing strategy document and supporting implementation of TB elimination mission in the state of Kerala and the Union Territory of Lakshadweep. He is also a Bernard Lown Scholar in Cardiovascular Health and visiting scientist at Department of Global Health and Population at Harvard T.H. Chan School of Public Health, Boston, USA. Welcome, Dr. Rakesh. Thank you both of you for agreeing to be the chairs for today, and over to you both.
Good afternoon. Thank you for giving us this opportunity to be here. Am I audible?
Yes, Varinder, yes, you're very much audible.
Thank you. And we are indeed looking forward to this meeting where, old people like me with gray hair at the verge of retirement look at vivacious new generation who are the future. And I've been asked to talk about of my journey in 20 minutes and then also give the direction, which itself is a big lesson for me, how to encapsulate something like that in 20 minutes, but I will try. But before that, I would request Dr. Rakesh to make his opening comments and then we'll take it further.
Right, sir. So good afternoon, everybody. It's a privilege to be co-chair with Dr. Varinder Singh and be talking to all these young scientists today. So I remember a few years back, I met a mother, her name was Priya. She was narrating a story. She was talking that her child was feverish and losing weight, and she was constantly tired and unable to play with friends as before. She could not understand what was really happening to that child. And at the clinic, they told her that she needs a TB test. And she was narrating how difficult it was to get a test done, where a tube need to be inserted and something need to be injected to get this sample for that particular test. And this was so frightening. I mean, the frightening or the pain that they all underwent was she explained that thing very clearly, and it was still in my mind how she narrated that. And when the test came back positive, and still she found out that she need to take pills for over 6 months. And then she realized that the pills will not come in the form of a syrup or chewable tablets at that time. They need to crush the pills, all this sort of stuff. And child was away from school for many days and nobody was willing to even play with the child. Imagine how you also feel when knowing that your child has gone through all this difficulty because the child got TB from you. The hundreds or thousands of mothers or fathers are facing this situation every year if they are fortunate enough to have access to diagnostic facilities. And in many places, children with TB might be dying without even knowing the reason. Across the globe, nearly 650 children, they die from TB every day. 80% of those before they're reaching their 5th birthday. Children cannot raise their voice. They can only cry. The only person whom I saw raising for children, as far as TB is concerned, is Dr. Varinder Singh. I'm really happy to have you, sir, as the chair and the first speaker today. I'm really excited to hear your journey because you are the best person who actually raised your voice for children with suffering from tuberculosis. Thank you so much.
Thank you, Rakesh. I had the advantage of riding on the shoulders of the seniors who had been working in this field for long, and we all learned and carried on the torch. It's not only me. Yes, I was at an opportune time when the program was getting involved. I was at the right place at the right time, so I could make noise. But you will be surprised to know, initial 5 years, I was told, hang on, we are still struggling with adult TB. Don't talk of pediatric. We can't even think. It took me 10 years. And in first JMM, there was one bullet point on pediatric TB. And then for next several years, I struggled to say that pediatric TB cannot be put under special conditions like HIV.
Varinder Singh, lady.
Sorry. May I continue?
Continue, sir. Please continue.
So it was that it was under special conditions, and I said we are not a special condition. We are part and parcel of this country. The children form 20% of the growing population, and they cannot just be shoved under special conditions for few lines. Well, it took me another 10 years to get a separate chapter on pediatric TB in the national guidelines. We have many country first in the world, that we had the first FDCs pediatric formulations available. We were the first country to introduce that. We were the first country to have national guidelines on pediatric TB. They went through several evolutionary steps, but we were the first. So, yes, I do bring that perspective to you. And when I was told today to talk on challenges and gaps in pediatric TB diagnosis, uh, is my screen visible?
Sir, you can put it in slideshow. Yeah. It's visible now, sir.
Yeah. So I thought if I just talk to this August gathering on what tests I have used and now they're useful or not useful, sometimes it's good to have no baggage of history, particularly when disruptive technology puts all that you have been using in the past, like PPD, like BCG test, something which should not be talked of today. So we are at a cusp of knowledge or technology where the way things are done has already changed. And there are challenges which are across right from health seeking behavior to identification of the uh, person or child who's likely to have TB, going on to initial investigations like X-ray, how to read an X-ray, chest X-rays. Now we also have computer-aided diagnosis, and then the fact that you have to have a confirmatory test, which is so difficult in children. And most of the evolution has happened around that. Two things which have disrupted the way pediatricians have been treating TB and they have to relearn was one thing, which is that there is a possibility of a microbiological diagnosis based on CBNAT now, which is rapid. And second is that now in today's time, we are talking of drug sensitivity directed therapy. We're talking of knowing RIF resistance right at the beginning. So profiling of patients on clinical basis, which was so difficult and which was so inaccurate, which led to delay of right treatment for a lot of patients, is something which has taken a back seat, and we have to do with the newer tests which are able to give us RIF resistance upfront along with a microbiological diagnosis. However, it's not easy to forget history when your new tests are not so perfect. And your new tests are able to diagnose only 50 or 60% of the possible TB cases. Then you have to go back and learn from the past lessons, learn from the masters of the past, like me, to know. So today, when I talk to this August gathering, it's not reminiscence. I'm not going to talk more about what we used to do. I intend to talk more about what I has been my learning so far as to what is my wish list. What is the missing piece which is not there? I'm not going to talk about excess or operational challenges, because these are young scientists here who are going to spend next 20 minutes with me, and I want to spend time with them to give them an ask related to what a pediatrician feels that is required by the patients who have TB. So that's what is the usual ask, be it that of a doctor or an individual or a community. We all talk of need to eliminate the disease. So for an individual to eliminate the disease from the body, for the community or global level disease elimination from the world is something which we're looking at, for which we need tests to diagnose and we need treatment to treat. But if you look at opportunities to treat or eliminate disease, they are not only lying with those who have the disease. They start from exposure. Can we decrease the exposure by early identification of the contact case, or by better hygiene, or better cough hygiene within the household, or which is basic hygiene taught to a child? From those simple steps, after exposure, when somebody goes into TB infection, in tuberculosis, you have a window of opportunity to prevent, to prevent this going to a disease state. So is it a possibility infection which is subclinical, which may not come to your notice for any symptoms, but can still be picked up because the person is exposed? Are there tests? Because there are interventions which are available as a prophylactic or preventive therapy to prevent this infection going to disease. And within the disease, whether we can identify the disease correctly, give them optimized therapy. So gone are the days when the national program started as RNTCP, we had category 1, 2, 3, where you had optimization of therapy more based on whether you were likely to spread the disease, you were category 1. If you were not likely to spread, you were category 3. And if you were not understood by the program because they could not fix your resistance pattern at that point of time, you were category 2, you got an additional drug. To today's time, where there is a possibility with a rapid test to know which one needs treatment as MDR versus first line therapy. And that's a big change or disruption. How do we do that? And finally, to know an endpoint of disease that you are actually cured. So can I have a test which will give answer to most of these? Or do they need to be compartmentalized? Can I have a test which can identify those infected but not diseased very clearly from those who are diseased among children, where almost it goes like a continuum in terms of their age? Are there tests to identify those diseased correctly, efficiently, and specifically? And are there optimized therapy available and to know an endpoint? This is the ask a pediatrician would have, a clinician would have from the newer generation. Look, I need tests to answer these questions, to serve these patients well. But I need these answers as a point of care test. This is something which is doable where the patient is, not that I have to send specimen, difficult to get specimens to a distant lab and wait for a report after several months. We need an answer then, a rapid test. And that's something which is important. And that's something we have also learned from a recent pandemic of COVID. Because if you look at the potential resources and human effort which was required and which could be recruited, this was tremendous. This has been a great learning. Though COVID badly affected the TB services in our country, but it also perhaps can compensate by opening our minds of the global health community to deal with TB as a challenge with same zeal, where everybody thinks of, okay, new test, new therapy, efficient test, catching it fast and treating it fast. Is it possible? And one thing which it very clearly emphasized also was a need for excess and efficiency in service can come when we can somewhere kind of, make it less fancy, make it closer to the patient, in the hand of the patient. So you have to take out the charisma out of it and make it a triage test which the patient can do at home by themselves, like a urinary pregnancy test or like a screening test for the COVID, which anybody could do at home and see whether they were infected. That's a good triage test which was available. And if it comes positive, go for confirmatory test. So we actually need to change the way we think if we have to go further. And that is why I'm not going to delve more on what I have learned in my life. I think it's already detailed. The immediate task which is there at our hand is to look at two categories of tests: the triage test, which will identify who in a pool of at-risk patient may actually have the disease. So what should be the character of this triage test? The character of this triage test would be having a high sensitivity, so high probability of having TB, and a triage negative patient should typically not require further investigation. So that degree of specificity is also required. It shall be great if it can distinguish infected from diseased, pre-symptomatic from symptomatic. That would help us put somebody on preventive therapy. Why it is important? Because in most communicable disease, be it COVID or TB, the fact that you have been exposed is something which was very important. So if you have been exposed as a child to someone who has got TB in the household or in your school or wherever you work or go, spend most of your most of your time. If you're not diseased, you may be infected because you were exposed, and that's what prompted the access to care. And therefore, having a test which could answer this question would be very good, because there is a efficient preventive therapy which is available. Obviously, this test has to be efficient to allocate patients to receive critical, yes, expensive confirmatory tests which are available. So this testing would be required both at community and facility level. It has to have a high sensitivity, but specificity will be a key determinant for its effectiveness and cost saving. So it cannot be poor specificity. It cannot be like a single test for HIV. Now we use triple tests. That okay, you bring out all and then confirm, because here the numbers required to be tested are much higher. So most current triage activity involves around the symptom screening, which is subjective, which misses asymptomatic or minimally symptomatic cases. And it's vulnerable to poor quality of care. And that's what needs to change. Then we also need the post-triage test to identify people that have TB, to provide a positive result to justify starting a patient on treatment, and identify the best suited treatment, drug sensitive or drug resistant. Which drug are you resistant to? Currently, the confirmatory tests like NAT, expert or TrueNAT or culture methods are able to give this answer. They are primarily still done at distinct district centralized laboratories or at a DTC level. They're not available at most primary health centers or closer to community. And despite there being a great leap from smear, which had a very poor yield, less than 10%, this test has definitely increased to 30 to 40% in a general term, or maybe higher in older children who have more open disease compared to the younger children. The major challenge, even despite having these tests, is need for a suitable specimen and access to specimen. Unlike an adult who can cough out and give you sputum, a child, even if it is reducing sputum, cannot volitionally give you a specimen, particularly a younger child. They may tend to swallow it more often or may not have a cough which is forceful enough to bring out that expectorate sputum. And therefore, there's a need to collect it. Either the swallowed sputum from the stomach as a gastric aspirate, or an induced sputum through the nasopharyngeal aspirate, or a swallowed sputum which has been passed into the stool and digested and passed, where you may find the mycobacterial DNA, or maybe a swab from the mouth. But there is a need for an access to specimen which varies. So if this test has to be a point of care, are there any criteria to define these? So that the criteria are given as assured that it should be affordable, it should be sensitive, specific, user-friendly, rapid, robust, as much as possible, equipment free or requiring minimal equipment, and deliverable to those who need them closer to where they are. And to this, some child-specific things may need to be added. So a point of care triage or confirmatory test is something which can be done in preferably in a single clinical encounter, provides you the result on the same day without requiring special technical expertise, so that you are able to decentralize it, and it's suitable for all. What do we mean by suitable for all? It is not only all ages. TB is a disease which has very varied manifestations. So what about extrapulmonary? What about quasi-bacillary disease? It should be a test which should be suitable to all. Unfortunately, just like the Holy Grail, it remains elusive. It's difficult to find a point of care test which meets all the requirements we do not have. People have tried to give target profiles, product profiles for tests which you need for triage, how they should be able to efficiently identify those who are likely to have disease, whether they have symptoms or not, or they have risk factors. That's your triage test. That we should be able to do it at a community or a village level with a very good accuracy of about 70 to 80%, and time to result which is not more than half an hour, preferably about 5 minutes, and for which there is an internal quality control, and it's an affordable test. A confirmatory test should be able, or possibly a rapid biomarker based test, which can identify maybe an infection from disease, which can identify amongst those trials as those who are truly diseased, again, at a very high sensitivity and specificity, which should be in the tune of more than 65% sensitivity, I would say more than 80%, and specificity, which is more than 95, 98%. That's what you need. Are there such tests available? Well, they're not. This is the profiling. This is what we need. Our current tests, be it a culture, smear microscopy, a cartridge-based or a chip-based test, the indigenous TrueNAT or TB lamp or a lateral flow assay, none of these are anywhere close to being a point of care test, answering all the requirements, while some of these, particularly the NATs and PB Lab, it has been possible to decentralize them to some extent, but not as much as SMARE, are more efficient, are more sensitive, are more specific, but they're not doable for all kind of diseases. They are not doable for all kind of specimens without pre-processing, which again shifts the usage of this test away from the community. And that's the challenge we have to meet and see what can be done. So the WHO endorsed tests today, some of them are pretty rapid, like cartridge or chip-based tests. You can get answer in less than 2 hours, but then these are modular tests, and a machine capacity is limited. If you have one machine in an 8-hour or a 16-hour shift, you are still able to do not more than 15 or 30 tests in a day. So in a high burden area, as today the diagnosis of TB is concentrated to district hospitals or larger, where we have more footfalls with sick children, there is actually turnaround time is huge, despite this being a rapid task, and may run into days to weeks at some places. So that is something which we have to think of when we are creating a rapid test. Can it be a non-modular, non-dependent on lot of machines? What can be done to make it rapid, truly rapid at a deliverable side, or the cost is so less that more than one machine can be bought for the laboratories or facilities which have high footfall. That is the additional need which I feel as a clinician, because it's very disturbing that you are waiting for 7 or 10 days for a diagnosis because you want to now be sure whether it is RIF sensitive or resistant. In the earlier days, which was less efficient in a way, but you could start treatment early on, and because it was more clinical and you were not dependent on others. So there is some loss of value or some loss of time as we graduate to one from one technique to the another one. Though I would still say the value addition by NAT is huge over the clinical scenario, which I was alluding to. When it comes to drug susceptibility, we have limited further, because LPA is one of the easiest tests which has so high limit of detection that it's unsuitable for a large proportion of pediatric specimens, extrapulmonary as well as pulmonary. Cartridge-based tests in our country today, we only have available for rifampicin. So we do not know about INH, which is the another elephant in the room. And as yet, the second line XDR panel is not approved for usage in the country under the program. So that remains another challenge, because when you get products which are created for diverse situations, sometimes they are not so well localized that they provide the answer. So there is a big gap between ethinomide resistance and iron resistance picked up by the XDR panel vis-a-vis the phenotypic panel within the country. And that's what has been kind of holding back the usage of these tests under the program. And these are the other challenges which we have to keep in mind when you youngsters are going to develop a new test or fine-tune the existing test. It's not important to redo the wheel. It may be good to make it more efficient and look better. For a child, the assured criterion user-friendly also has to be child-friendly. And what are those child-friendly things which we need? The children are not many adults. So the child-friendly test is which is easily accessible, which is available on demand, which does not hurt, which is acceptable both to the child as well as to the caregiver, and which is universal. So this is something which is very, very important for us to remember, along with the assured criteria when we are creating such tests. So my ask finally is a test which meets both of these criteria. And we have no such test which meets all of these criteria and provides answer for all of these stages of disease which we see today. New. A lot of this has been asked in the past. So what is there already there is the in the try test, you have an aero nose which can look at the volatile products coming in the breath and tell you whether this individual has actively multiplying bacteria, tuberculosis bacteria or not. CRP is a distinctor between symptomatic disease versus non-disease. People have even used artificial intelligence to analyze cough, whether the cough on a TB patient is different from cough in pneumonia or wheezing or cough or other causes. If there is some things to suggest that you may be able to pick up, and that would be quite interesting, because then a recording of cough could be made on the phone and sent to the machine, and you could see whether do an AI analysis, no visit required. Wow, fantastic! If it had the sensitivity and specificity which you need, but at least a good try test is possible. So that's something a lot of work has been done on. The other difficulty is, even for X-rays, be it digital or otherwise, the interpretation is very difficult. So we have computer-aided diagnosis, which is going on. It still is little away from being very efficient. It is as efficient or as inefficient as a doctor at this point of time, a non-radiol, a clinician, in my viewpoint. It needs to be sharper than that. Then you have this new test which talks of the expert TB host response. So where you're looking at host response to TB blood messenger RNA on expression of 3 differently expressed genes, which are expressed differently when you have disease. So it can discriminate between active TB and other diseases. So this is something which has been being talked of, may come in future, but it will only tell you that you have active TB. Will not provide you answer as to whether you have drug sensitive or drug resistance. So till I have a treatment which is not dependent on drug sensitivity, this test becomes limited in its utility. It is easy. It's a blood test. You can do it very fast. The access to specimen is not difficult. I am not sure it can be decentralized to that extent. But the difficulty would be, once you have diagnosed TB, you're back to the same question. You still have to access this specimen. HIV co-infected, but as yet has not been demonstrated to be so efficacious for non-HIV co-infected children. But this is something which may come up in future, because easy to collect urine and easy to do lateral assay. Whether the existing test can be better optimized without, so people are now looking at whether you can use NAT for an oral or tongue swab or a stool or an aerosol collection, which does not require a lot of extra pre-processing. So that, because the moment more processing is added, you need a technical person, you need a lab, and it shifts back away from the community to the larger centers. The difficulty is to marry this convenience of having it right at the community. So it has to be not that technically intensive or so automated and so easy to run, so less dependent on other support, that you are able to use in the periphery. A battery supported device which has an internal quality check could be one of those answers. People are also looking at cytokine assays, and they're also looking at activated the T cell activation markers in the TBSA, like CD38 based TAM test, which could differentiate between the infected and diseased. So am I raising a list which is not doable? Am I raising questions which are like in a domain of a fantasy or a science fiction? Well, if you were in the era of 1940s, when there was LJ solid medium, who would have thought of a vector? That was at that time a fantasy or science fiction. So the dream of yesterday
Sir,
Teacher, what?
sir, we cannot hear you. Sorry, sir, we missed you in between.
Oh, I'm sorry. It seems that I may have a bad connection. I'll switch off the video.
Yeah.
Is it okay to continue where I am, or you would like?
Yes, sir.
Okay. So what I was saying is that in the past, what we did and what we do today, it looked as if it was a science fiction. It was something which was not possible, but today it is possible. So what Becktech or MJet is able to do today was unthinkable in 1940s. To have a test which will tell you in 1.5 hours that you have rip resistance, even in as late as year 1999 or 2009, I was not able to think of a test like that. It was not available. And today we use it every day. So it is possible to dream as long as we have intention to put foundations under those castles which we have dreamed. And that's the reality of tomorrow. And that's what I am hoping this current generation will provide. What they need to learn is two lessons. One is try to find a test which answers many, if not all. I don't think any single test will be able to answer all the questions which a clinician desires to be answered in the case of tuberculosis, yet a large majority of them could be. And try to maximize the efficiency of that test in a way that it is possible to bring it. You know, the problem is once the technology is taken up and it's a disruptive technology, the further efficiency cycles are not fast, they're rather slow, because the commercial angle of extracting the cost which has already been invested comes first, rather than going for an upgrade. Upgrade comes once the saturation with the initial product happens. So that brings a kind of a gap. It's not simply a commercial thing. It is also a lot of work goes in to develop a to create new things. But they can also perhaps work simultaneously. Once you have it, a good should not be an enemy of best, but at the same time, good should not make us forget about the best. So it is very important to keep that perspective in mind and keep on making this test more efficient. At the same time, think differently of a new test which would not have any of these limitations. So a human response test or a biomarker is a disruptive technology which will work differently. So if that can be married with a therapy which is not dependent on drug resistance of the child, but this is more related to a host response which would be created. So instead of an antibiotic, you are using a biostimulant to kill the bacteria. Wow, fantastic. All of these tests will become irrelevant. You don't need to know whether they're resistant or sensitive to A or B. All you need is, okay, because we know it is possible. Why? When there was no drug available, our body was able to heal 30 to 40% of TB by itself. So the body has capacity. What is that missing link? What is it which we have to understand, whether we can have that intervention to create that mechanism which the body has to stimulate that mechanism to take care of the disease is something we need to work at. So these are the thoughts I leave you with as I come to the end of my talk, that we need to think progressively like that and not try to go back doing algorithms, which again we have guidance which has come. Okay, look at guidance where chest X is available or not available. Yes, that's reality, that's the real life. But I refuse to accept this reality when India is on moon. I will be on moon the day a feasible test available in the countries is available at every corner and is not only available at a district. That is the day I am on moon, or most of the clinicians will be on the moon. Yes, we celebrate the effort made by our scientists to reach moon. I am not undermining that at all. It's just that as a cliched statement, or I'm just drawing parallel using those words to say that for us in our field, we will reach moon when we are able to create a test which is efficient, which reaches the periphery and delivers, rather than trying to use inaccurate tools in more inaccurate algorithms and trying to bring objectivity. That somewhere takes us away from our right path. As a public health expert, one may argue that you till the best is not there, shouldn't we do what we have? Yes, but for last 40 years, that strategy, or for last 70 years, that strategy of leading edge, let's do what is doable, has somewhere made us tardy, has made us slow. We haven't made enough progress in the field of health. And therefore, such algorithms can only be a stepping stone, cannot be the answer. We have to find answers which are doable, where our imagination will carry us to the worlds that are going to be there, because even though they are not here, but without that imagination, we will go nowhere. Thank you very much.
Thank you so much, Dr. Varinder Singh. As expected, I was expecting that you will talk for the children, and rightly you have talked for the children. To the research community, now we need to clearly understand the challenges which Dr. Varinder Singh was telling to us. It's on us also. We also need to, there are so many challenges there, but we need to, yes, so many progress have been made, but we have miles to go. So any questions to Dr. Varinder Singh at this point, you can ask. Anybody? You can ask directly.
So any of the audience present here, if they have, they can raise the hand and come in. There's a question and answer.
The question is basically, it is being written by Shakil Ahmad. You have transferred dream to the right population, the next generation. Hope they would come up with the solution soon. Stool as a specimen has done some of your dream. In our publication, we could get gene expert ultra on stool to be 83.3% sensitive. What is your thought on this? The question is about gene expert ultra on stool.
So, hi, Shakil, good to hear from you. So, this tool looks like an answer, but the difficulty is with ultra, an interpretation of the trace results in a high burden country. So I do not know the sensitivity which you have. I read your paper, but the sensitivity you have reported, how many of them were traced. But this tool brings two questions. It is a very fantastic area, and it's a specimen which is very easy to give, though still not on demand, but still possible. So that's something which is very good about stool. But the difficulty so far has been about the concentration mechanisms and its adaptability. Though now we have 3 simple methods which have come up, which can be used, and that's an area which looks exciting. And if something comes through the aerosol collection of a crying child, just or a talking child, or a child who is singing, and you collect the aerosol which is created by the child, and if you could find a signature there, would be fantastic. That won't hurt him at all, and that not everybody loves to lug the stool around, so that may be easier. And then I also am very excited about oral swabs. That's something which can give us answers, but current sensitivity of the available WRT is not good. Given the reported sensitivity, if what Shakil is reporting can be replicated all over at ease, and does not raise many trace results, things would be much more easy. Because the moment a trace result with rifampicin resistance comes, then it opens another set of difficulties and questions, and creates a hindrance. So, but yes, it's definitely a good step. Thank you.
Varinder, is the organizers allowing me to speak a little bit?
Yes, please, sir.
Oh, thank you very much. Varinder, it's always a pleasure listening to you. And the stool result here was that it was positive with the 83% among the positives, which was we have done on sputum induction, those cases. And you are correctly said that the trace results are the challenges. And we have tried to have answer of that in our next publication. next research, which will be published soon. And there, what we have done, that we haven't treated a group of trace results who had improved while they are in the hospital clinically. Fever was gone, the cough is gone, the child was feeling better. We stopped treating them. We have not started treatment on them, and we followed up them for about 1 year. And none has came as TB in that 1 year. Though we know that for TB, 1 year is not the ultimate answer, but still we can tell that these are at least not subclinical TB. Maybe they are incipient TBs. We don't know. So that's the area where we have landed currently. Thank you. Thank you allowing me to speak a few words.
Yeah.
And especially when Varinder is there, I love it. Thank you, Varinder.
Thank you, Shakil. Before we look like a mutual admiration society, I'll just quickly add, it's just a respect for a very diligent worker which Shakil is at Dhaka. So the difficulty with trace results is not only whether they are what to do with whether to treat or not to treat. The difficulty is that the quality of specimen itself can give rise to trace results, even in a diseased patient. So that distinction, it is easy for several things to be done by experts like us in testing areas. What is my dream and what has been my challenge in life is that whenever I have tried to replicate what we have learned or done in our setting and have gone to a wider area, the result goes on tremendously, because that zeal, that efficiency, that degree of expertise is not transported along with the test. And that's a challenge where when you are trying to create a test for the whole country, which comes. So that's my concern. Over to you, Rakesh. So if there are no more questions, should we invite the next speaker?
Yes, sir. We can go to the next speaker.
Okay, so the next speaker is Dr. Aparna Mukherjee, who is a senior scientist, medical and in charge of clinical studies and trial unit at Division of Developmental Research in the ICMR. Dr. Mukherjee has graduated and postgraduated from Kolkata, and thereafter she was working with Dr. Kabra's team as a PhD scholar at All India Institute, Delhi, when I first met her. And she has been a very diligent scientist and now is looking after the division as a senior scientist, the Division on Clinical Studies and Trials. And she is going to take us through the diagnostics of pediatrics. So over to you, Aparna.
Thank you, sir. It's always a privilege and honor to speak after you. And I guess what the topic that I was given was like newer advances in pediatric TB diagnostics and challenges. It's something very similar, but what I would like to do is just go into a little bit more details of some of these newer tests that are already in the market or are being tried. So I'll share my screen. Hopefully you can see my screen. So
Yes, Dr. Aparna, we can see.
yeah, thank you. So what I would like to go through is, like sir gave a very nice overview as to what we have and what we desire to have in order to properly diagnose and treat children with TB. So I would just take you through what the current approach is, what are the challenges, and what do we need newer tests, what are the ones which are in pipeline, and what do we actually need to detect. So one is obviously LTBI, which is kind of now we are coming to the state where we want to treat LTBI, so we need to detect. We would also very much like to see which of these kids would actually progress to disease, that is, we would like to actually detect the incipient disease. And of course, diagnosis is always a challenge. And if we did have something apart from our clinical acumen to see the response to therapy, that would be good. And drug resistance. So this is our wish list of tests that we want. So in India, as we know that childhood tuberculosis is something which is there. And as Sir said, Dr. He and his team, along with Dr. Kabra and their team, they have worked relentlessly to bring childhood tuberculosis in the notice of all policymakers, to state that it is an important problem which needs a separate understanding and deliberation. But in spite of that, the recent report that was brought out by RNTP has stated that there might be a gap of around 55% in TB notification. So that gap may be because of people, one is that people are not notifying, but which is unlikely because TB notification right now is quite robust. So the more probable reason can be that we are not actually diagnosing them when we should. Even globally, it has been seen that in case of pediatric tuberculosis under 15 years, a large chunk is missed out. So one is that they suffer themselves. Two is that they increase to the burden of transmission. So if we are to improve the case detection, so what are the advances that we need is, or that we have already made in this field is, one is with the efforts of all our seniors, there is improved awareness amongst people treating tuberculosis, people treating children, that PEDS TB has to be taken into account. We have good tests like Expert, even Expert Ultra now, which can give us a result much faster and with a higher sensitivity and specificity. As you said, stool has been approved as one of the specimens in the WHO document. It is a good specimen in the sense that it is easier to collect than a gastric lavage. But yes, there might be more studies needed and more in-depth research needed on this. And there are some other non-sputum biosamples like urine, which are proving to be interesting and may help us in diagnosing. But the challenge is that we don't have anything which is like a gold. We do have gold standards. We don't have anything which gives us all our wish lists. And in children, the problem is that when we are talking of a gold standard, when we take a reference standard, we always need to take a composite standard where we will have a large chunk of smear negative or culture negative kids. And host biomarkers and the other cares are all things that are in the pipeline and we hope to see in future. So current approach to diagnosis, definitely clinical presentation, clinical judgment is the most important thing, backed by microbiology, radiology, and other supportive tests. So clinical features, I don't think I need to repeat, but they are kind of non-specific. So you have to be very good in judging them, having a high threshold of index. Again, on the other side, the pitfall of this is if your threshold of index is right, you are suspecting TB in all cases where there is no weight gain, the slight fever, you may actually end up over-diagnosing TB also, because we do see cases where anti-TB drugs are almost used like, some kind of a supplement. So, but clinical features are the most important thing which any diagnosing physician should look for. So, as we all know, it's unremitting cough or fever for more than 2 weeks, losing or not gaining weight for the last 3 months. And if you get a contact history, that's well and good. If not, not. So this is again the scoring system that has been underlined in the WHO document. And as Sir said, scoring systems, we have been trying various kinds of clinical scoring system for a long time, but they don't seem to have that kind of a specificity in order to ascertain that yes, this is MTB. So whereas you do rely on your clinical judgment, and when you see a particular child, the symptoms and signs can lead to a lot of diagnosis, but totally relying only on these scoring system may not be a very good idea. So we come to biospecimens and diagnosis using these. So what are the specimens that we usually collect for our kids? So it is, of course, they cannot cough out sputum, as sir said. So we have gastric lavage or gastric aspirate. Induced sputum is something which was very much tried in various research studies in order to induce the sputum with the help of hypertonic saline, and then and actually make the child cough, and then either take a nasopharyngeal aspirate, or if they could cough out sputum, that would be collected. But again, this has its own pitfalls. Then we have bronchoalveolar lavage, but then you need a setting where bronchoalveolar lavage can actually be done, and it can never be in public programs. Stool, as everybody is referring to, is the newest, most interesting specimen, and we yet have to see how it helps us in the long run. So this is basically a comparison of ambulatory GA and induced sputum. This is one of the study where Dr. Varinder Singh was also one of the PIs, and where we compared gastric lavage with induced sputum. And gastric lavage, though it's a more invasive procedure where you're actually putting a tube, and you do need a setting as well as some amount of expertise on the part of the nurse or health personnel who is carrying this out, but it does have a higher yield than induced sputum. Be it in smear and culture. At that point, we hadn't done gene expert. Next is bronchoscopy. If you are a pulmonologist or if you are a pediatrician who is conversant with this, there's nothing like it, because you can actually see the lesions or the nodes. You can see the compression of airway, and you can collect the sample from those particular segments. But again, this is a very center specific modality which cannot be used widespread. Similarly, EBUS can also be used if you do have the facility and the expertise. And if you do have it and you keep on adding it, the cumulative yield, of course, will go up. When you start with only GA and induced sputum, if you have a cumulative, say in 100, you start with 100 children and you have a cumulative total yield of 34 using only the conventional gastric aspirate induced sputum, you add bronchoalveolar lavage or you do an EBUS, goes up to 75. But that depends upon the centers. So these are the specimen collections. We'll come back what we do with these specimens. Before that, of course, chest X-rays, the bread and butter of diagnosing TB, and we know these are the general features, either mediastinal lymphadenopathy, miliary shadow, or cavities. But again, diagnosing TB from chest X-ray in a child becomes more difficult than in an adult, because you don't always get those very typical cavities and lesions that would point towards TB in a child. You may actually get lymphadenopathy, which would be difficult to diagnose, differentiate from other respiratory disorders. That is why it is difficult in these children. If you're not sure, antibiotic is advised, and then repeat the X-ray to see if there is an improvement. So this is the RNTCP algorithm. Sorry, I'm still calling it RNTCP. It's the NTP algorithm, which states that if you have any of these symptoms with or without contact with TB, an X-ray needs to be done in the child. If the X-ray is normal, fine. You can still check for EPTB if you have a high suspicion rate and you think that there are lymph nodes or there is some other system which is affected, do the diagnosis accordingly. If there are non-specific shadows, then we can give an antibiotic course and see, and then redo the X-ray. If the X-ray is highly suggestive of TB, then we go forward and do a specimen collection, which could be either expectorated sputum, gastric lavage, or induced sputum. And then instead of going for smear culture, we directly go for a nucleic acid amplification test. If it is negative, we again look for some other sites, which is, they may be associated lymph nodes. As lymph node tuberculosis is very, very common in children. And we can repeat a sample and reevaluate the case. If NAT is positive, so we get a microbiologically confirmed TB, which may be drug sensitive, may be drug resistant, and we treat accordingly. So this is the algorithm that is given by our own program and usually followed by everybody. So now when we collect the specimens, what do we do with it? We used to be doing expert RIF, which would give us the positivity for the organism as well as give us a sense of the resistance to rifampicin, not INH, only rifampicin. So now we have ultra, where most of the guidelines are saying directly to go to ultra. The advantage is that it has multiple loci detection, and so it has got an increased sensitivity. But the problem, as Sir referred to, so this is just more of data which just tells you that yes, expert does have a good sensitivity as well as better specificity actually, whether you take culture as the gold standard or you take a composite gold standard of cultured and clinical radiological features. And irrespective of whether you take sputum or gastric aspirate as the specimen, you do get a specificity somewhere close to 90%, above 90%. But of course, your sensitivity varies. So it's good with gastric aspirate, it's good with sputum as well. But if you consider stool or nasopharyngeal aspirate, the sensitivity does fall. So that is something that we need to keep in mind. And there is a repeat of this. But as Sir said, the concern with expert ultra is a lot of trace reports. So why the reason that is being given for these trace reports is that it could be because the bacillary load is less in that child, or in case of HIV positive child, or an EPTV where the pulmonary load is less. But still, this is a gray area as to whether they should be treated or not. WHO recommends that treat these children. Specifically, look at their clinical condition, judge their symptoms and signs, and treat them. But if you get a trace report, the rifampicin resistance definitely has to be reconfirmed. We do not know whether that person is resistant or not. So this is a concern that needs to be ironed out, smoothened more as we go forward. Then we have TrueNAT, which is based on the same philosophy and the same science, but then this is indigenously developed nucleic acid amplification test. It is portable, and so it's battery operated portable, so it can be taken to peripheries. And we have now studies which have compared TrueNAT with culture as well as with gene expert with culture, and it does show a similar sensitivity and specificity like the expert when compared with culture. So this is another alternative which we now have, an indigenously marketed alternative. And the counterpart of this is from expert groups is the expert Omni, which again is a battery operated portable instrument. But not sure how much it has reached or it can reach the PSCs as yet. Now, if we come to the non-sputum specimens, which is the target of most of the researchers now, because for children, we would prefer a non-sputum specimen, would make life much easier to collect and to diagnose. So one of them is urinary lamb, which has been a lot of studies have been going on urinary lamb for quite a long time. Right now, we do have some studies which states that the sensitivity can range anywhere from 33 to 57. 33 is not a sensitivity which we can accept. 57.89 still, since we are accepting a sensitivity of around 50 for stool. Specificity for this is high, and this is more important for the HIV positive children if you want to use it. So then if we come to the other tests, as I said, that there would be two types of tests. One where we would want to triage and see that this child might have TB. And then the second part is where we want to confirmatory test. For triaging, or we do have some digital X-ray and then deep learning or used on it. So we have these softwares from various companies which gives a automated reading of the X-rays. But how useful are they, and have they been validated in children? So if we see there, so these are the three ones which are available in the market. And if we see their sensitivity, specificity, sensitivity seems good enough, but it decreases when the person is HIV positive, if the TB is a smear negative TB, or if there is a history of TB earlier in that person. So taking these into account, it may not be as helpful as it is intended to. But yes, an automated reading would help, because if we can have it in the peripheries, we would not have to rely or depend. We still have to rely on our physicians, but maybe if we do not have somebody to read, this is a perfect way to counteract that. But these have not been validated in children as yet, because reading the X-rays in children are much more difficult than in adults. So we need the validation in children. Then, as I said, that post-transcriptomics, again, there have been, plethora of work on transcriptomics. People have been trying to pinpoint which ones will help in diagnosing TB. And these are 3 transcript signatures given by Sweeney et al., which expert has incorporated. So these are basically genes modulating the inflammatory pathway, and they might be able to distinguish TB infection from TB disease, basically diagnose TB disease. And this is a prototype where you are using finger prick blood test and measuring the expression and giving a score. And it was tested against expert ultra in one of the published literature and gave a sensitivity of 87%, the specificity of around 94%. So obviously much more work is needed on this, but it might be a good test in the sense that we are using a non-sputum specimen, a finger stick specimen, and so it might be helpful if it is validated further and we are sure of its sensitivity and specificity. Then we have some cytokine-based assays. Again, these are all in the prototype phase. Aerosols, where we are wanting to detect MDA, and MTP, and we are also using our nucleic acid amplification test. If we can use these specimens, that again will be very helpful. Then we have a battery of confirmatory tests, which are in the prototype or in the testing phase. TAM-TB, which is T cell activation markers. Basically, we want to characterize activation markers. So these 3 activation markers as well as some activation markers are characterized here. It can detect TB. It has sensitivity as well as specificity, but again, it will never become a point of care test. Then you have some cell-free DNAs, which may circulate in specimens like urine. So if we can actually make a test out of this, that will be valid. People are looking into other specimens like tongue swabs, oral swabs, because tongue papillae may have a concentrate of MTB, which can be detected. So this is again a promising approach. For drug resistance, whole genome sequencing is a way where we will be able to detect all genotypic resistance. But again, how far it will reach the periphery is something which is absolutely debatable and to be seen. So this is something that I have taken from one of the borrowed from one of the papers. So for triaging and for confirmatory, what is it that we have now in terms of, in terms of in terms of what is in early prototype stage, in late prototype stage, and which is like people are going through a further validation stage. So as we said that cytokine based tests, cell-free DNAs, these are in a very early research method. They may be helpful, but they are still not anywhere where we can actually start using or start testing them. Late prototypes are, we had expert HR, which is the three gene signature for transcriptomics. We have the tongue swabs, the LAM tests, so which can proceed further. AI-based X-ray, a lot of work has been done on this, but then we need to do it on pediatric TB to be able to say that we can use it in children. Stool, again, a lot of work has been done. A lot of work has been done from the participants of this particular workshop itself. And so this might be, getting included in our clinical practice very soon. So yeah, we would want a confirmatory test which is rapid, which probably is biomarker based, and which can diagnose both pulmonary and extrapulmonary disease. And we would like to use a non-sputum, non-invasive specimen, and like to get the decision within the same day or at the most the next day. So these are all aspirational definitions of an optimal confirmatory test. But yes, as Sir said, we are confident we will get a better diagnostic test as we go forward. So in a nutshell, it is a challenge to diagnose childhood TB, but we've come a long way from earlier, and we have molecular tests now. We will have better molecular tests and other specimens. So we need to keep in mind in our DD that yes, we need to suspect TB, and not only suspect TB and not start treatment on a presumption of TB, but actually diagnose it, confirm it, and then start treatment. That's it. Thank you.
Thank you, Aparna, for that fantastic talk, which took us through the journey of what is there and what's there in the pipeline. It's kind of echoes and gives some credit to the thoughts which I elucidated a little while ago, because your wish list seems to be matching mine. That means we are thinking in the right way, and let's hope there are more answers to it. Are there any questions which anybody wish to ask? Dr. Aparna, I'm sure will be.
There is some questions in the chat box, sir. One by Pranati Das. Does a trace report on expert not indicate RIF sensitivity status? This was the question by Pranati Das. So we may need to explain a bit about trace report in an expert.
Yes, a trace report in expert is when it does not give you a rifampicin resistant status, and you need a confirmatory trace, because here you're not getting a positive signal for all the loci of the genes that it is testing. So it might suggest that yes, there are MTB in that specimen, but whether it's rifampicin resistance or not, for that you need to repeat or get a other test.
There's another question by Dr. Rutuja Patil. Is TrueNAT recommended for diagnosis in children by NTEP?
I think Dr. Varinder will be able to answer this more, but TrueNAT has been validated by the program, and it is there in many of our peripheral centers. But if it has come in the documented guidelines, that will be able to.
Yes, it is very much part of the NTEP. In fact, there are more TrueNAT machines today than we have CBNAT, and there are equivalent tests. There is no need to retest a specimen if it has been done on TrueNAT with CBNAT or vice versa. They're considered equivalent. You may have other reasons to repeat a test, but do not repeat a test with CBNAT just because it has been done with TrueNAT. Thank you.
There's one more question by Dr. Satya Mahapatra. Is stool test recommended by NTP?
As
As
yet,
far
no.
as I know, not yet. Sorry, sir, please go ahead.
No, please carry on. So as yet, no, simply because the efficiency of testing is largely with ultra and not with the classical CBNAT. And under the program, we have as yet not accepted the ultra tests for the reasons which I alluded to earlier, which is the trace results. We heard from Shakil based on the studies that says they did not treat. But then the specimens, if they are homogeneous, and you test the specimen, and because the test was not, the specimen was not homogeneous, you may not have enough bacteria in the tested part, and you may get a trace. You can't believe that trace result to be only reflecting a non-disease status. So therefore, it is a signal which adds a lot to the noise.
Yes, sir, I agree to you, and I would think that it is the clinical judgment which should add on to that report to actually decide whether you treat or not.
But we do need to talk about it, and we do need to work more in it, because our country has a significant proportion of people going to private sector. So ultra is available in all private laboratories. It's used by most private institutions. So sitting at a National Center of Excellence, when we get questions, queries around that ultra, we still have to respond to them. So while the program may not have accepted it for several reasons in their wisdom, but as we have a dual responsibility. One responsibility is to support the government and the program. The other is as a pure researcher or as a clinician, and to guide the the overall profession. So it is the academicians who have the responsibilities, of the researchers who have the responsibility to guide the clinical way a test is being used, be it for private or public sector. So our responsibility therefore has to go across the spectrum and cannot be only restricted to NP.
Exactly, sir. Dilesh has asked a question: how is the stability of TrueNAT? Ma'am, you have any clue about stability of TrueNAT? I mean, even the question is not clear to me.
Yeah, I'm not sure what do you mean by stability of TrueNAT.
Dilesh, you want to speak? You want to ask that question directly?
Yeah. I mean, like initial studies, there was some field level instability issues.
The storage of chips is something which is important. Otherwise, there are challenges with both WRDs, whether it is expert or there is CBNAT. The expert machines would not function if you do not have temperature control. So at very high temperature, they will go back. So interruption is in supplies. So the stability around the procedure is affected by several things. The advantage with TrueNAT is that, okay, you have battery operated systems, and they do not always require as much temperature control as is required by the CBNAT, so the cartridge based. So there are subtle differences, but one of the things which has happened in the pandemic is that both these machines were also, because this is basically a PCR machine, which is either cartridge based test or a chip based test, and depending on what probes you have added, you can test it for n number of diseases. It's not disease. The machine is not specific to TB. The chip is, or the cartridge is. So these were the machines which were also used for COVID. So that has kind of made people more familiar with their functioning, and it is now more easily understood by more number of technicians at the periphery thanks to COVID. So that has been an advantage because of the pandemic, and now those difficulties are little. Thank you.
There is one more question in chat box by Sebin Abraham. Is testing for HIV routinely recommended by NTP for all children diagnosed to have TB? Dr. Sebin has asked this question.
Yes, that's right. Irrespective of the age, HIV, because of the co-infection of this disease having its own implication, this is a reflex testing for TB in every HIV patient, and reflex testing for HIV in every TB patient is something which is a national policy and is recommended, is done.
But I think we have answered all questions. Do we have further questions from the audience? Like, feel free to ask because we have time. And we have the experts also with us. So any more questions to the experts?
So one question which we frequently encounter regarding children transmission of TB to children is that if the mother is having TB, is it safe to breastfeed the child who's born to such a mother? So even though the guidelines say if on treatment they can do breastfeeding, still there is a lot of resistance from the community to kind of breastfeed the children. So I just wanted to hear thoughts of Varinder sir on that. Like in your practice, have you found that difficulty to convince them?
You see, there are few reasons why mothers would not feed their children when they are suffering from TB. One is that the general ill health itself, they're too sick themselves. So that something may require a temporary cessation or less frequent breastfeeding. That's one issue. The second could be where there is a myth which is very common, that the disease may spread through the breast milk, and they say iska to doodh hi kharab hai, and that's how there is more stigma related to disease, and that's where we have a definite role to say no, this is not. The breast milk is never a reason for transmission. Transmission is by close contact. So if the child is going to stay with the same mother, she's going to be the primary caregiver, then her cough hygiene, airborne infection control practices are more important rather than feeding. Feeding actually would protect this child from many other diseases which will come up because the child is given top weight. So that's something. If it was not that the mother is moribund, is too sick, or sometimes if the mothers have a totally drug resistance TB, that there are no drugs available for a small child, then it may be prudent to isolate and separate. We are not talking of stopping breastfeeding. Then we are taking a very, very bigger step, which has its own implication. But that's when you are dealing with a situation that mother has an infection for which you do not have you would have nothing to feed. So you do not want to risk this child, and you therefore may isolate them. But that is a very rare situation. I would say more talk theoretically than I would have really seen, but I would not say it never happens, but it's very unfrequent. So as a nutshell, it is that breastfeeding is not a problem at all. And if the concern is about the lactation, the drugs being there in the feed and that having an impact on the child, no, none of these drugs are secreted enough in the milk to cause any effect or side effect in the child. So neither that drug can be used as a prophylaxis preventive therapy if your child needs preventive therapy, which every newborn whose mother has TB needs. That will not come through breast milk. That requires separate treatment, preventive therapy, once you have ruled out the disease. And neither is it enough to cause any side effect in the child. So therefore, breast milk feeding is absolutely safe.
Thank you, sir.
Usually, sir, like I have a question to you. So usually we know that there is most of the national TV programs, they report just half of the cases. And there are so many reasons cited, including what all you mentioned about sensitivity of the test, lack of appropriate test, point of care test, all this sort of stuff. One important challenge, another challenge is about the specimen collection, especially with regard to children. So we need to get a good quality sample while doing gastric aspirate or something. So is it okay? I mean, I'm just asking your experience on shifting this task to a nurses at the periphery. I mean, is it possible? Like, is it advisable of collecting?
Yes, indeed, it is possible. You see, we talk of task shifting. We have shifted injections, we have shifted diagnosis to taking a definitive decision to treat or not treat to staff nurses. And here is a test which is much more simpler. It's yes, it does require some degree of training. It does require some degree of protections for the healthcare workers because you are creating an aerosol. But it's not something which cannot be done. In fact, if we have to devolve this test to the periphery, that is where we have to spend our effort on to train them. And it's not difficult. It is possible to do. It's a relatively simple procedure.
Right. So operationally, I think for improving access, like we can actually try to train the staff at periphery, especially the clinicians or the staff nurse at periphery, and try to do that. I mean, I'm just putting for some questions for the researchers listening to this, for like especially who are interested in implementation research or operations research. Most of the biomedical research questions Sir has actually posted to you. So these are some of the operational or some of the implementation research we can put forth. I was also thinking about this collection transportation system. Like, for example, many of the times Sir was mentioning about as a clinician, we expect the result in a day. So most of the times what happens is, even though the machine's turnaround time is just 2 hours, we may get the results after 7 days or 8 days. So why is that delay? It's not a technical delay most of the times. It's an operational or a managerial delay that is happening in the field. Somebody need to collect the sample, the person may need to travel, transport that, reach the facility. In the facility, the machines can work for 24 hours, but most of the times what we have seen is it will work for just 4 hours or 8 hours in a day. I'm just putting for some other questions. Say, for example, if we can deliver food at the doorsteps, like by Swiggy or Zomato, within 15 days, if we can get a taxi like Uber in 3 minutes. So why can't we plan a specimen collection transportation system across the country if that is a major challenge? I'm just putting for some questions. Or else, how best we can use all these machines lying with us? Is the machines good if we use it in a PHC, or is the machines good if we use it at a district level with a good specimen collection transportation system? At the district level or at the block level, where we have 24 hours, we can run these machines with minimum HR. Only thing is, we just need to transport the samples using a hub and spoke model, using this kind of models. I'm just thinking which will be more effective, which will reduce the turnaround time. So reducing the turnaround time is something which we are actually looking at. These are some of the operational questions which the researchers can think of. Sir, your thought on this?
So I would entirely agree with you. That's the reality. So there are examples where people have, and practices where people have ensured transport mechanisms, and even drone has been used in the hilly areas. But whether consistently or it's one-time use is something which one has to look at. It has to be consistent. But if from my understanding, I would be very happy if somebody would develop a auto loading cartridge system for GeneXpert. You see, unlike TrueNAT, where you have to first take out DNA, then put into the chip, in GeneXpert, if you have a cartridge which is charged, if it can stay stable for a long time, fine. You do not have, you need 4 lab technicians at least to run 24 hours lab, 8-hour shifts, one person of minimum 4 or 5 people. Most labs have only 1 lab technician. That is why the machines run for 8 or 10 hours, because that after their working hours are over. Now, if the machine was so automated and so less, it's once the cartridge goes in, there's nothing a technician can do about that test within the machine. So the machine is totally automated. The technician is only required to charge the cartridge and load the cartridge. If that loading and unloading could be automated, your machine will continue to work 24 hours. No? You charge 30 cartridges, put them in a carousel and attach it to the machine. Once the first 4 are done, next 4 are loaded, they will be done. You don't need a person. So that's the kind of innovations we need. Okay, this far it was done by Cepheid. Why not create a cartridge carousel by our mechanical engineers? So we have to think how to make the technology more efficient and how to maximize the benefit out of it. And there are several thoughts which I'm sure many people have, at least I have, which I'm happy to share.
So we got 2 more questions in the chat box. One is by Karun Sandeep Visa. What is the gastric aspirate? What is the sensitivity in CBNAT versus TrueNAT for the gastric aspirate? This is first question. And what is the advice in programmatic conditions, whether to opt for CBNAT or TrueNAT? and gastric aspirate sample should reach within 4 hours time to the NAT test. How this could be improved in a programmatic conditions. Any thoughts on this, Dr. Aparna, Dr. Varinder? GA, the question is about sensitivity for gastric aspirate using CBNAT and TrueNAT, and whether it is better
Similarly,
TrueNAT
that's
or
what
CBNAT.
the WHO document says when they were given approval. So to that extent, they have worked similarly, but I'm not aware of head-to-head comparison between these two tests to know. They should be concordant, but I'm not aware of many tests where in a large enough sample where two have been compared head-to-head.
Yes, so there is one more question from Gulham Khader. It's from Afghanistan. The child makes 22% of the total TB cases reported to NTP. So that's a scenario there. 22% of the total cases are children. What screening and diagnostic test you recommend in this setting? The TB incidence is 189 per lakh population. Resource limited, unavailability of radiologist. All these are the scenarios. So what will you recommend here? This is the question by Gulham Kader. Maybe we can attempt, try this, answering this question.
Yeah.
We don't have the complete picture of there, but still, sir, you want to take this question?
Okay, so
in a high incidence setting?
Kader, this is not something which was not there in India or parts of India, where the access to a trained radiologist may not be there. So what can work in this situation is teleradiology. I have worked with one NGO, which is UK based, who is working in Afghanistan, and read the X-rays on their behalf or seen their difficult cases, not routinely, which tells me that it is possible. Yes, it has its own challenges. If it is not a digital X-ray, it's an analog X-ray. You take a picture, the quality of picture sometimes is not so good. It has its own challenges. But teleradiology is one answer to that, which can do it, and which but to my mind, while you use teleradiology, what you should be using is developing local capacity. And it should not be left only to radiologists. That's what we have done in India. Our training module has largely initially focused on how to read X-ray chest. I used to spend half an hour to 1 hour in every training only on reading of chest X-ray. So the clinicians have to learn. Ultimately, whatever the radiologist writes, you have to interpret in light of the clinical picture and have to agree or disagree with him. You are not just a passive recipient of an opinion or a report, because you have the film with you. So building those capacities is also important. So while that may be difficult given the difficulties in your situation, but I think you would have to have a multi-pronged approach where possible train local capacity, where not possible use teleradiology till you are able to build up the local capacity. And thereafter, the teleradiology can be your quality assurance mechanism. So somebody makes a diagnosis, once you have trained them locally, that goes to by teleradiology to the next person, and he says agrees or disagrees, and that's how it gets sorted out. So those are the mechanism which some of which we used. We did not use as much of teleradiology, but we did use a lot of training material, and that's something we are still working on, creating a module on chest radiology with focus on TB. We've created a lot bit. Maybe that's going to be my first job after I retire.
The last question for this session by Dinesh Kumar. Which one, according to you, is the bottleneck towards TB control? Is it case finding or continuum of care? Which one way higher?
I think case finding weighs higher than the continuum of care.
Yes.
Because the availability of drugs, with the ease of availability of the drug, it is more of case finding difficulty. But I would also add here, case in our zeal to fill up numbers. The moment we go into case finding business, one difficulty is we start following a hypothetical target. Obviously, it can only be a guess. It's our estimates. And then the target filling approach leads to diagnosis and treatment of many who actually do not need treatment for TB. And they are the one who will not continue treatment. And that's where your results of and your effort will get diluted, because you are working with a larger chunk. So one has to be very wary. We must build the quality and quantity should be like a Siamese twin. If they are not twinned together, then one will suffer on the account of the other. That is.
Totally agree, sir. I mean, we need something like target to identify how many cases are being missed, but it's not like we need to pressurize people on meeting those targets to so many false cases will come up during
Target
that.
is for planning. That is a very
Correct.
important tool for planning.
Correct. Great, sir. So shall we move to the next session now?
Yes,
Yes.
please.
So next session is by Dr. Aishwarya Venkatraman on TB vaccines. So Dr. Aishwarya Venkatraman is a pediatrician by training. So works as scientist E at ICMR, NIRT, National Institute of Research in Tuberculosis, Chennai. Her areas of interest include pediatric infectious diseases and immunology, pediatric TB, vaccines and malnutrition. She also leads the pediatric TB vaccine trial at NIRT. So this is a very important and relevant topic that we are going to listen now on TB vaccines. Over to you, Dr. Aishwarya.
Thank you so much for this opportunity to talk about the vaccines which are available for TB. What I'll be talking now is the already existent vaccine, which is the BCG, which we all know, and what are the new vaccines which are in pipeline, and where is the research up to. So shall I just share my slides?
Yes, please.
Can you all see?
Yes, Aishwarya, we can. If you can just, yeah, perfect.
Okay. Okay. Fine. So as I said, it is the topic is about vaccines in tuberculosis, and I'll be talking what are the vaccines which are currently available and how far we are in terms of research on TB vaccines. So with any kind of infectious diseases, we know that vaccines go hand in hand. And with TB also, what we are looking for is a vaccine which can prevent the disease, prevent the infection, and which will also prevent the recurrence of TB. So what we have currently now is the only one vaccine which is available, which is the BCG, which I'll be talking in the next few slides. So why is this TB vaccine really important? So what we are aiming with the current WHO TB strategy is to reduce the incidence of TB worldwide, especially in the TB endemic areas. And what we aim to do is, with the introduction of either a new vaccine or a vaccine which has been repurposed, or with new drugs, we should be aiming to introduce that around 2025 so that we can see a greater effect in the reduction of TB incidence worldwide. So with the vaccine, it is a part of WHO TB strategy as well. And what we are aiming is to contain the disease, TB disease. I'm really sorry with my voice, but hope it's audible quite well.
Yes.
So what we are looking for in a vaccines, the attributes of ideal TB vaccine, we want a vaccine which is very safe. At the same time, it is highly efficacious in all age groups, in children, infants, as well as in adults. It is effective against all forms of TB, both pulmonary TB, extrapulmonary TB. It should be effective against both drug-sensitive and drug-resistant TB. It should be something which is easily producible, as well as it is easy to store. We should be able to produce in large scale, and it can be transported to all the remote setting where it can be used for vaccination. So that is why we are looking for logistically feasible. And we also need to have a vaccine which can be administered in situations where we have very low technology, or we have underdeveloped countries as well. So this is what we are looking for, an ideal TB vaccine. So according to WHO also, the ideal TB vaccine has been defined. So this document came out in 2018, where they defined what exactly they are looking in the TB vaccine. So in terms of preventing the pulmonary disease, both in adolescents as well as in adult population, we should have a vaccine which has got an efficacy more than 50%. In preventing the TB disease. The protection should be at least 10 years or more after the first vaccination. And we should not have repeated doses of boosters. The dosage should be maximum of three doses, or a booster dose which is needed only after every 5 or 10 years. So this was in terms of looking at the prevention in pulmonary TB. Again, in newborn and infants, an ideal TB vaccine would be something which offers more than 80% of vaccine efficacy. I'll talk about why we are looking at more than 50% in pulmonary TB and more than 80% in neonates and infants in the next coming slides. But in terms of having efficacious vaccine in preventing TB disease in newborn as well as in infants, we need to have a vaccine which is more than 80% efficient in preventing the disease. And that should prevent all forms of TB, that is pulmonary TB, extrapulmonary TB, including TB meningitis and disseminated TB. So what are the different types of vaccines which are available? So we have 4 types. But for the first one is the attenuated live vaccine. That's where the BCG comes. We have the inactivated vaccine, and then we also have other vaccines like subunit vaccine and the recombinant live vaccine. So these are the 4 types of vaccines which are used for the development of TB vaccine. So coming to the age-old, the very well-known vaccine, which is the BCG, the Bacillus calmette-guerin vaccine. This is the only vaccine which is licensed and approved for TB. We all know it's a live attenuated vaccine, which was developed by Albert Calmette and Calmette-Guerin. And this is a vaccine which is given as one shot at birth in most of the countries. And there are very few countries where they give it later in their childhood and or an adolescent age group. We know it is very effective against prevention of disseminated TB and central nervous TB. So this is where it comes that the current BCG vaccine, it gives an efficiency of around 80% in preventing disseminated TB and central nervous TB in young children, that is in newborn and children less than 5 years of age. And it kind of gives a variable protection against pulmonary TB. So this efficacy varies somewhere between 5 to 80%. It ranges from every country to country. And there are various factors which are involved why it gives a variable protection in different places. So this is what we have currently with our BCG vaccine. So this is history of BCG substrains which we currently have. So the vaccine which was produced somewhere in early 1920s. From that, we have actually come a long way, and the late strains of vaccines is what currently we are using. So what in India we use is the Danish strain, which is one of the late strain. We also use the BCG Moscow strain. Though it is an early strain, it is still being produced. That is where the Serum Institute produces the Moscow strain. So these are the 2 strains of vaccines which we use in India. So we can see that there are multiple strains which have been produced all over the world, and they have been used in different countries. So different countries use different strains. So how does this BCG vaccine work? So the BCG vaccine actually has got various roles. It not only provides specific immunity against TB disease, it also has got non-specific immune responses. And that is where it gives protections against non-TB diseases. So BCG is also known to protect against non-TB respiratory diseases. It is known to reduce the mortality in sepsis and infections like malaria, leprosy. And also it has been shown to be efficient against infections like Staphylococcus aureus and Candida infections. So how does it work? So once the BCG vaccine is given, at the site of injection is where the BCG starts acting. So at the site of infection, the innate immune response starts to work. So the BCG, once given, the monocytes, macrophages, and the dendritic cells, which are present in the innate immunity, they actually interact with the BCG, and they actually initiate the innate immune system. So while this is going on, this is where lots of cytokines and chemokines are released. And once these activated cells are ready, they are presented to the adaptive immune response to stimulate the adaptive immune responses. So they act as antigen presenting cells with the activated BCG inside, and this is where they go and stimulate the lymph node. So these cells, dendritic cells, when they enter the lymph node, they trigger the adaptive immune cells, and they are specific to the TB. So they produce the anti-TB CD4 and CD8 cells. That's when they release the increased interferon gamma and the granzymes, various cytokines are released. So this is how the anti-TB effect of BCG works. So we have also recently come to know that BCG not only stimulates the innate and adaptive immune system, it also stimulates the humoral cells, which is the B cells, directly by increasing the production of memory cells, plasma cell, and antigen-specific antibodies. So these are the studies which I want to highlight. So we can take the pride in presenting that India was one of these countries which did the largest BCG study called the Chengalpat study. So this was a large community-based double-blind randomized trial which presented the findings of BCG vaccination in the population. So more than 2 lakh participants were involved, and they were randomized to either receive BCG or placebo by random allocation. And we used 2 strains of BCG vaccine. We used the Danish strain as well as the French strain with 2 doses. The low dose where we gave 0.1 ml, and the high dose where it was given 0.1, high dose was 0.1 milligram and the low dose was 0.01 milligrams in each strain. So all participants were followed up for 15 years and by resurveys every 30 months. The first paper, which came out in 1999, that actually showed that BCG offered a low level of protection in children, and it didn't offer any protection in adults. So this was around 27% with a confidence interval between -8 to 50% in children. Subsequent to that, we had revisited the Chengalpat trial, and we recently produced the paper in 2022, where we followed up the participants who had the BCG vaccine retrospectively, and we found that BCG revaccination in community offered a modest protection, around 36%, against the development of TB disease after another 15 years. But since it was a retrospective data analysis, with this finding, it actually needs to be further evaluated and validated in a larger study. Subsequent to these 2 studies and other studies, we also have a meta-analysis and systematic review, which came out last year by Martinez et al., which actually highlighted what are the shortcomings of BCG vaccine. So this was a study with 26 cohort, around 17 countries, done across 17 countries. And it's actually found out that BCG is very effective in all forms of TB in children less than 5 years. And it did give a significant protection that includes TB meningitis as well as disseminated TB. And it also gave protection in contacts. These contacts were defined as those who had tubercular skin positive. They were defined with interferon gamma, the IGRA assay positivity. And BCG vaccination protected against the disease in contacts as well. The overall effectiveness of TB, of the vaccine against TB was around 18%. So what this study actually highlighted was BCG vaccine is very effective in preventing TB in young children, but it needs a booster dose in older population after 5 years of age. So subsequent to that, we have had studies which have renewed and reinstigated the BCG revaccination in children as well as in adults. So 2 major studies which were published recently are the studies which were done in South Africa and another study which were done in India. So the first study is the South African study, which included adolescent between 12 to 17 years of age. And they had given revaccination to this group of children. These all children were TST negative or quantiferon negative, and they were all HIV negative. So this study compared BCG with a newer vaccine, which was H4IC31. And what it found was both vaccines were equally good, but BCG was slightly better in reducing the rate of sustained conversion with an efficacy of 45%. So this was one of the landmark study which actually said that BCG revaccination is actually very useful. We also have a study from India, from Indian Institute of Science, by Annapurna et al., where BCG was given as a revaccination to healthy adolescents as well as and adults, to young population between 18 to 28 years. These were all HIV negative, TB exposed population who are working in a TB sanatorium. And though it is, they could not clinically follow up these participants, they did an immunological study on these participants where they said BCG revaccination significantly boosted their antimicrobial Th1, Th17 responses, both in IGRA positive as well as IGRA negative individuals. Subsequent to these studies, we have various studies across world which have started BCG revaccination. The countries which are doing are South Africa and Brazil, which are taken up BCG revaccination to look at the prevention of infection, prevention of disease, as well as prevention of recurrence. These three vaccine will be looking into these three components. We also have study from Australia where they are looking at the effect of BCG on COVID-19 and healthcare individuals. So this is a study which will be coming out very shortly. So as I said previously that India was one of the foremost participants in the BCG trial initially. So now also India has proposed to do a large scale, community-based study where we'll be looking at BCG revaccination, both in adults as well as in adolescents. So the adult study will be taking, starting off very shortly in the next couple of weeks to months. Subsequent to that, we'll be taking forward the adolescent vaccination in the community. This will be again a revaccination where we'll be looking at prevention of TB disease as well as prevention of TB infection. So other TB vaccines which are in pipeline. So this is still how I spoke about the BCG vaccine. So how the TB vaccine development pathway works is like initially we discover the target vaccine, what can be used as a vaccine. And then it goes into preclinical development where we have the animal studies. Once that has been done, it comes into the clinical studies in human population, which includes phase 1, phase 2, and phase 3 studies where the vaccine is scaled up and validated. Once that is done, we have the vaccine registration and the launch of the vaccine in the community level. So these are the TB vaccine pipeline at different stages. So what I'll be focusing here is the phase 3 vaccines, which are 5 of them, which have crossed the preclinical, clinical, as well as the phase 2 vaccines study. So now we have around 5 vaccines which are at phase 3 level, which I'll be talking in my subsequent slides. So starting with the phase 3 trial, the 3 most advanced vaccine candidates are the MIP, the VPM1002, and the M72 vaccine. So MIP and the VPM1002 vaccine is the vaccine study which we are currently studying in India, which is already ongoing. The MIP vaccine is a whole cell vaccine, which is heat-killed intradermal vaccine. And the VPM1002 is a recombinant vaccine where it is a recombinant BCG vaccine. Again, it is given in intradermal. So these 2 vaccines are currently being studied in India as a multicentric phase 3 trial where we are comparing this vaccine against placebo. And we are looking at prevention of infection, prevention of disease, as well as prevention of recurrence. The M72 vaccine is the other most advanced vaccine candidate, which is a protein and adjuvant subunit vaccine, but it has to be given intramuscular. And it is a planned phase 3 trial vaccine where 26,000 individuals need to be enrolled between the age of 16 to 32 years. It will be a prevention of disease vaccine. And currently we have the preparatory study evaluation going on. So this is the study about M72 vaccine, which was done in South Africa, Kenya, and Zambia, sorry, where 3,000 odd plus adults were recruited, and the vaccination was given to 18 to 15-year-old. It was a randomized trial with 1:1 allocation. They either received the M72 vaccine or the placebo. And these participants were followed up for 3 years to look for the incidence of TB disease. And what they found was the vaccine efficacy at the end of 36 months for around 50%. And this they they also included the immunology component in the vaccine study, where they found that the M72 specific antibodies and the frequency of M72 specific CD4 T cells were highly increased after the first dose, and it was sustained throughout the study period. The vaccine was quite safe because both in the placebo group as well as the vaccination group, the incidence of adverse events or any kind of immune-mediated disease were almost similar. So the VPM 1002 and the Imuvac vaccine, which I told, which India is currently doing, is an ongoing study where we are looking at the efficacy of VPM 1002 and Imuvac in reducing the incidence of TB disease. It's a double-blind placebo-controlled study where we are recruited healthy household contacts of new smear positive pulmonary TB cases. The study age group is anybody above the age of 6 years, and the participants are followed up to 3 years. So the study would be completed next year in March 2024. Sample size with the 12,000 individuals. So currently the last part of the study is going on. So these are the other two phase 3 trial vaccines, which is NTB-VAC, which is again developed by Bharat Biotech, which is a genetically attenuated MTB vaccine, which will be given intradermal, and it's a live vaccine. And the study is currently going on comparing MTB-VAC with BCG in South Africa, Senegal, and Madagascar. So GAM-TB vaccine is a Russian study, which is a protein and adjuvant subunit vaccine to be given subcutaneously. Again, they are looking at vaccination in adult population for the prevention of TB disease. So these are the list of phase 2 vaccines. Once they have finished the phase 2, they will be coming up to the phase 3 study. So these vaccines are again in the pipeline, which will be coming in the next 1 to 2 years as a phase 3 candidate vaccines. So again, the phase 2, we have quite a few vaccines, around 6 to 7 of them, which will be coming as a phase 3 vaccines in the next few years. So phase 1 vaccines are also underway, which again is either vector-bound and mRNA vaccines. I don't want to go too much in detail because they are only in the initial stages of clinical studies. So coming to what are the challenges which we face in development of a new TB vaccine. So we know that TB immunology is something which is evolving. We are still currently trying to understand the immunology of TB. So we have a large gap in the knowledge of immune system in terms of both adult as well as in pediatric TB. We do not have a predictive TB marker where we can say that this one marker can be used to define the disease as well as the development of infection to disease in any patient. So lack of any clear predictive marker as well as no good correlate of protection is one of the challenges which we face in TB. And for any vaccine from the animal study, it has to come up.
Thank you so much, Chris.
To know 4:30.
Animal study.
Yeah, I know. It's okay.
Neeraj will make to decide because those guys are going out of.
Okay,
Dr.
that's fine.
Dr. Christine, you must be muting.
Yeah. So for any study, any vaccine candidate to be used in the human population, it has to first be have a preclinical studies and then have the clinical evaluation. So what happens in TB vaccine is we do not have a suitable evaluation in the animal model. And we do not have a credible preclinical evaluation indicator to say that this can be definitely used in the human studies. So because of these, we face challenges in development of a new TB vaccine in the human population. Coming to the economic side, the demand and market uncertainty also plays an important role. And in any vaccine trial, what is most important is the funding, which is also a big challenge with TB vaccine development. And for any vaccine development in infectious diseases, it has to be tried and tested in a setting where the incidence of the disease is quite high. And with regards to TB, unfortunately, we do not have a very good infrastructure in TB, many of the TB endemic countries, like in African nations where the infrastructure is not up to date or not enough to try out a very large scale clinical trial. So how do we overcome these challenges in developing the TB vaccine? So currently there are lots of consortiums which have formed across the world who are all the scientists and the researchers are coming to work together to develop a new vaccine or a better vaccine or try the revaccination of BCG. So even WHO has established a TB Vaccine Accelerator Council, which wherein all the researchers across the world will be working together to develop a neo TB vaccine. So with that, we hope that we will be able to address the TB elimination and we will be able to address with the WHO and TB strategy. Thank you. Any questions?
You can unshare the flashlight.
Yeah.
There are some questions in the chat box, Dr. Aishwari. Is recombinant BCG vaccine tested in pediatric population?
One second, I'm just trying to unshare. Sorry, I'm just not able to.
No, you have to click on the share screen button below and stop share.
Okay, no, I'm just not able to enlarge my Zoom screen. One second. Sorry.
Okay, button.
No, I'm sorry, I'm not able to do that.
No issues.
Yeah,
Dr.
that's
Aishwari.
fine.
Yeah.
I done it. Yeah. Sorry.
So is recombinant BCG tested in pediatric population?
Yes, they have included children above 6 years of age, and but I should say that the number of participants is probably quite low to validate that vaccine in pediatric population yet. But the study population includes children above 6 years of age.
In background of rampant infection in India, is 80% efficacy look over ambitious in Indian trial in HIV population?
I don't think so. We have any studies to say that whether we are ambitious or not. We may be ambitious, but this 80% of efficacy, what we are looking at is in neonates and children, because we already know that BCG works approximately 80% and more in that age group. With regards to pulmonary TB, what we are looking at is only more than 50% efficacy in reducing the pulmonary TB in an adult population, because in India, that's not what we are achieving currently. Above 80% is something which we are looking in neonatal and in young children population less than 5 years of age. I would not say it is over ambitious because we are yet to try it out and see whether it's ambitious or not. Without trying, we can't answer that question.
Yeah, so there are other questions also. Say, for example, regarding Arun Kumar has posted this regarding BCG vaccine. Most of the child, they do not develop a scar. Is it okay, or is it due to not able to administer dose intradermally?
If you don't get a scar, most likely it is the technique of giving the vaccination. So normally, if the technique is current, a child should definitely develop a scar. But if a child does not develop the scar, despite waiting for approximately 9 months to 1 year, in my opinion, I think it is no harm to revaccinate the child. I would probably ask Dr. Virender Singh to address this question better than me. But in my opinion, if a child doesn't develop a scar even after 1 year, I think the child should be revaccinated.
I have a slight difference of opinion there, Dr. Aishwarya. The studies have shown, one, with time, the scar may disappear or fade down, and sometimes it is not seen later on. And some children just do not scar for some reason. It's not always the technique. Some children just do not scar. And people, when looked at the LMI, I mean, they did in vitro tests to look for whether there was any hypersensitivity developed or patient had benefited from immunization, there was evidence to suggest it. So revaccination, just because the scar failure is far more common. So it is not recommended. Yes, if you aren't sure where it was given, or there were issues about storage, there is no harm in revaccinating. Revaccination, you have alluded to some benefits. One of the things which is important to remember for revaccination is that this is our learning from past when BCG test was used as a test. So repeat usage of BCG always gives to a much bigger reaction and a larger scar, which sometimes can be very painful and difficult for the child. So revaccination, if you are sure that there was a difficulty with the primary vaccination, go ahead do it. Otherwise, maybe not. But this current evidence, if it is validated that revaccination increases your protection, then okay. If you're not seen a scar, it is better to be vaccinated. But I think still that information is with us for sure. I would be a little hesitant.
Yeah,
Yes,
so
sir,
the
I think
Arunkumar's question was,
you're absolutely right that we first have to validate that BCG revaccination is definitely effective. So until that study comes out, you're probably right that we should be slightly cautious in revaccinating.
yeah, so there are further questions on this. Up to what age child receives good protection post BCG?
So I did highlight that in one of my slides that we had the systematic review which came out last year. So up to 5 years, there is good evidence that BCG vaccine immunity lasts. And after 5 years, it starts waning. We actually don't know until what age it actually completely wanes off. This we haven't studied yet, but yes, after 5 years, the efficacy of BCG vaccination does reduce.
Again, will booster enhance protection period? Any evidence on that?
Immunologically, I would say yes, because there have been certain studies which have done only the immunological studies to say that BCG revaccination enhance the immunity. But giving a BCG revaccination for prevention of disease is something which we are yet to completely study and evaluate in the field setting or in a research setting. So that is something which we have to do. But immunologically, yes, giving BCG vaccination, it does enhance your immune responses.
And did studies which look at revaccination mention how long the protection lasts? Do we need multiple boosters?
That is something, unfortunately, we won't be able to answer because I don't think so any studies have looked at the immunity of revaccination yet. I think there was only one Malawian study which looked at repeated revaccination over time. But in their setting, they found it didn't reduce incidence of TB. But that study also highlighted that the incidence of HIV positivity was quite high. So we just have one study which has looked at repeated BCG booster vaccinations, but not many studies are there to say that.
Despite almost 100% coverage of BCG, why pediatric TB is not coming down?
The coverage of BCG is 100%, but the efficacy of BCG is only 80% in neonatal period. So that is one of the main reasons that pediatric TB is probably not coming down. And so that is why we actually need better vaccine than BCG, which can give kind of almost near 100% protection.
Yeah, also what BCG does is also a bit, something which we need to discuss, right? So it's not actually, what is that actually doing? Is it preventing the infection, or is it preventing the disease, or is it preventing the severe forms of disease? So what is it major role that is actually playing? The incidence will come down only if we have a proper evidence on bringing down or cutting down the infection, or even cutting down the disease directly. So if we generally look at the relative risk in that way, or the protection in that way, we can explain all this sort of stuff. Any more thoughts on this?
The only thing I totally agree with you that we do not have evidence to suggest how much is the protection truly BCG offers for inhaled route or pulmonary TB. It does definitely have an impact on dissemination. But if the revaccination is decreasing subsequent conversion in adults, then obviously it has some role even in pulmonary TB, whether it is at the time of infection or from infection to disease is something which we do not know, which we know partly. So it's not used as a strategy to decrease the incidence of TB as a TB elimination tool, but it does decrease the severe form of TB, and therefore there are benefits. Like the countries who had withdrawn BCG from their immunization schedule did see more TB, did see more hematogenous spread, and many of them reintroduced BCG back into their vaccination schedule until they had kind of burnt out the adult contacts or the disease in the adult population, the infectious pool. So once they're infectious, still their infectious pool was maintained. And one thing which we're going to struggle in future, and since I'm talking to investigators here, is important to bring out, is we are forgetting the role of zoonotic TB. So even when the human form of TB goes down, whether vaccination, if it is useful, we'll be able to withdraw totally, because the zoonotic forms is something we are not even looking at or addressing. We don't even know what is the quantum of the disease, and we have no control for that. And primarily, TB came to human beings from it's a zoonotic disease to start with. So if till that source, where it is going to persist, is not removed, any disease we have been able to eliminate is only the disease which is restricted to mankind. Those who are out beyond the mankind, you always need to look at the other source where it may continue. And that's going to be a challenge with TB. So we'll be able to bring down the infection, but to eliminate, to get to a zero state of infection, well, that's a big challenge as of today. That's all from my end.
Sir. When we are thinking of moving to that level, we may need to address all the concepts of One Health, zoonotic disease, everything need to be brought in, especially in all settings. So there is one question which is unrelated to this topic, but again, since Sir is here, most of the practitioners still use Mando test to diagnose TB. How much significance it has in the current scenario?
So Dr. Aparna had shown you the algorithm, and the algorithm, there's no place for the tuberculin test, right? So that gives itself an answer. But I would also add something. That algorithm talks of how to deal with a microbiologically diagnosed. When it comes to clinically diagnosed case, there is a role for having a TST or an IGRA to say that this individual is infected or not. So if the patient is not infected, the child cannot have disease. So the negative test, kind of helps you to rule out the disease. But the difficult part is none of these is 100% sensitive or specific. Since they're not 100% sensitive, you do not know a person is falsely negative or negative because he's not infected. So again, one of the ways I used to say is that when you need crutches for your diagnosis, you look for TST. If you're sure of your diagnosis, you don't need it. And that's where it becomes difficult. It has a very limited place, I would say, if used correctly. So the strength, often we write for a tuberculin test and the lab does a test. God only knows what strength was used. And we have, that thou shall always be positive if above 10 millimeter kind of concept about tuberculin. But the response to the reaction in the arm is dependent on the antigen given. If you're given 10 TU, 20 TU, it will be falsely positive. So if less than 2 TU, not right with PPD-RT23. But there is no PPD-RT23 available anywhere in the world. So what we have in India is not a standardized product. And since we do not have a standardized product, we use it because it's an old friend. You don't want to get away. It's an old habit, which is difficult to discard. But its role is coming. That's all I would say.
When we think of revaccinating using BCG, so for example, say since this is a live vaccine, so in settings where, say for example, HIV is more or less, say for example, we don't know how it works in other immunocompromised conditions, especially in adolescents or adults. So are we planning to do HIV test and rule out HIV before giving the next dose of vaccines, or like how do we proceed with that sort of stuff in a programmatic setting?
In a programmatic setting, probably for research setting, yes, definitely we should be testing for HIV and doing it. But if you look at studies which were not studies, if you look at the BCG vaccination in African nations, they actually don't do HIV testing in many countries. They actually give the vaccination even if they, without even testing for them, because HIV is much more endemic there. But if, in my opinion, I think, yes, we should be testing them for HIV before giving it because of immunocompromised and getting a disseminated.
I think, questions are over for this session, sir. I think we can move to the next session, sir.
Yeah, I think that shows the right choice of the topics and the equally good choice of the speakers, me excluded. So, so well done, organizers. It's my pleasure now to invite Christine Ho. Dr. Christine Ho is the tuberculosis branch chief for the CDC India office since 2017. And she has a vast experience of dealing with this disease and its preventive aspect, particularly in US and globally. She has led a research consortium which focused on programmatically important questions related to the latent TB infection, particularly for children, and has several publications on that. So welcome, Christine, to the meeting, and we look forward to your talk. I'm sure we learn a lot. Thank you. Over to you, Christine.
Christine, you are not audible.
Okay. All right.
Now
There
you
you
are
go.
audible.
Okay, great. Thank you, Dr. Vinder, for that kind introduction. And it's, this is such a subject that's dear to my heart and I find incredibly interesting. So it's great to be amongst a group where all we're talking about is this. Yeah. So I think what I'll actually do though is I'm going to stop my video because the strength of the signal is not that great. Okay, and then I will go ahead and show the slides. Do you have them or should I put them up? How does that work?
Please put them up, Dr. Christine.
Okay, all right. Great. So let me put a slideshow. And I'm sorry I had to step out just for a little bit. Can you see now? I don't think so though, right?
No, not yet.
Okay. Oh, share. This is it. Okay. You know what? Let me actually take it off of slideshow. And then I think that's what I have to do. And then I'll put it on normal. Okay. Okay. No. No, it's going on. Hold on 1 second. I have to escape. End show. There we go. End show.
Should we project it for you, Dr. Christine?
Let me try one thing
Okay.
and then I'll do it. Hold on.
Sure.
Oh, share screen. Here we go. And then here. Yeah. Okay. And then let me make this a slideshow. I think you can see right now, right?
Yes, Dr.
Okay, there we go. Okay, very good. All right. So here we are. The discussion has naturally veered over to TB infection and TB preventive therapy.
Dr. Christine, yeah, it's your third slide and it's not in slideshow.
Oh, you know what? Why don't you show it? Because it's on.
Okay.
It's showing on the other screen. I have two screens, so it's doing something funny. Yeah,
Sure,
I will stop
we'll
sharing.
We'll do that. We'll do that.
Okay, thank you. Okay. Good. Okay, can you guys see the first slide?
Yes, you may start.
Okay, great, wonderful. So we're going to be talking about TB infection and TB preventive therapy and its role in our efforts to drive TB disease down. So go ahead and go to the next slide. So we're going to be covering a lot of topics, the importance of TPT, what is it itself? We used to call it latent TB infection, now we call it TB infection, the tests and treatment for it, the regimens, the concerns we have, what the programmatic experience is outside of research, and then research gaps. Okay, so I thought I would actually start with a case study. So just go on to the next slide. So like Dr. Rinder, I really started as a clinician. And so sort of sometimes the importance of some of these programmatic activities really hits home from the front lines as a clinician. So this is a case study. It's from the US, but it's actually just to show that even in the US where you think that you don't have TB or you don't have these issues are common everywhere. This is a 45-year-old white man. He was a realtor, businessman, very successful. He'd had a dry cough for a week. He saw a doctor who'd prescribed him a week of antibiotics, and he didn't improve. And he went on vacation. He went to a resort in Palm Springs, and he saw the doctor there, and that doctor said it was allergies and gave him decongestants and a cough syrup. And this is kind of very common, right? Even for us, we have a cough. We think, is it a viral thing, allergies, etc. After 2 months, he's still coughing. He goes back to the doctor. Now he gets doxycycline for 12 days. Now he has developed anorexia, loss of appetite, and he loses 5 to 10 pounds. And he's starting to have fevers and night sweats. So now he is changing his tactic. He goes to a doctor. They get a chest x-ray. And let's go to the next slide. So I know that everybody here is not a clinician, but what we can see from this chest x-ray is that the right bottom side of that chest x-ray, normally you see the heart outline very clearly and it's fuzzy. So, they call this a right-sided pneumonia. It's not a classic pneumonia. It's not like a sucked in, sort of one area, but they're not always like that. So this is his chest x-ray. Okay, let's go on. Okay, so he's given a stronger antibiotic, Augmentin, for 10 days, loses 10 pounds, still does not feel better. He's still coughing, has fevers, night sweats, classic symptoms, fatigue, shortness of breath. He can't sleep. Finally, he is hospitalized. And there's actually, I'm not even like putting the whole course on here, but sort of after a long delay and other sort of diagnoses that were considered and treated for, he finally gets a sputum smear for acid fast bacilli, and he's 4+ positive. So he has TB. He started on TB meds and feels so much better after 1 week. Okay, so as a clinician, you would think that that's it, right? But the question is, what else needs to be done? What other information do we need? We're talking about public health here. So we have to think beyond the patient and we have to think towards the community, right? So keep on going. Okay, so it is, go back. Yeah, so his TB is confirmed by PCR, and this is kind of a while ago, this case. So it was pre-Gene Expert, but we had an in-house PCR, and it turns out he had distant travel to a place with high prevalence of TB, to Cebu, Philippines, 20 years prior. He's married with 4 kids, and their ages are 2, 6, 8, and 10. Okay, so now go ahead. Okay, so now we look at the wife. The wife has been coughing mildly for 6 months. She has no other symptoms. Her skin test was done and it was 25 millimeters. And she is born in the US in a low prevalence area, and they get a chest x-ray. So next slide. And then you see this chest x-ray here. And basically, go back. It doesn't look it doesn't look bad. A lot of people would just call that normal. It's like a normal, healthy sort of woman. But because she is a close contact to somebody with smear positive TB, go ahead and move to the next slide. So they've gotten sputums. They're waiting for cultures as a pre-Gene Expert. She started on TB meds. And now the question, what needs to be done for the children? And now with WHO guidelines that we actually are going to be doing household contact and TB evaluations for all of the people in the house, right? Okay, so next slide. Okay, so the children are taken to a private provider who performs the skin tests and chest x-rays on the children. Well, the skin tests. So a 2-year-old has a 14 millimeter skin test, so clearly positive, way over 5 millimeters, never gotten BCG before. And the chest x-ray is read by the pediatric radiologist as normal. And we called them and said, you have to, this is when I was in the San Francisco TB program, we said, please bring the child in, bring the x-rays, and we can start window prophylaxis, which means that we actually will start TB medications while we wait for the results. Okay, so next slide. All right, so here's the chest x-ray. It was read as normal. And one of the things, I don't know if you guys went through this or not, because I had to step out, but the diagnosis of pediatric TB is kind of tricky, and even sort of pediatric radiographs. So chest x-rays for children can be tricky too. So this was read as normal. But what I would say, I don't know if you can see the pointer, probably not, but on the right side of the hilum, you can see it's very thick. You see an air bronchogram. You can see this, like sort of that right bronchogram really outlined clearly. Yes, thank you. And then when you go on the lateral, oops, go back, please. Okay, and the lateral behind the bronchi, you see this big fat kind of white thing. So that is not normal. That's lymphadenopathy. So he came, we saw these x-rays and we said, wait, this is not normal. Okay, so now's next slide. So if we had just relied on the reports, we would have missed this. So we had the chest x-ray reread. It was read as having right hilar lymphadenopathy. The toddler was started on 4 drugs, and we did not get further specimens for diagnosis. And I think this is part of that issue about it's, kind of hard and somewhat traumatic to get specimens from very small children. Okay, next slide. Okay, next child, the 6-year-old is negative, PPD negative, and the only family member to have a negative PPD, and he has no symptoms, and they get a chest x-ray. And we'll look at that in a minute. And it's kind of questionably abnormal, like what would you do? And I think this is so perfect what Dr. Vinder was saying before about when you use something like a skin test or an IGRA, quantiferin or T-spot, in the setting of trying to determine a clinical TB diagnosis. And the issue is, it's good if it's positive, it helps you. If it's negative, it doesn't help you that much, right? And we'll talk a little bit more about that later. So here, if you look on that lateral, so the back part, the posterior part looks fine, but look at below the Y, the branching of the bronchi. Again, it's kind of fat and it's splayed out. There's this big fat kind of opacity. And again, this is lymphadenopathy. So again, it's very easy to miss. Okay, next slide. So what happens is that mom's evaluation is going on, smear comes back positive. So we know that mom now has TB, and the radiologist rereads this x-ray and confirms there's that inferior lymphadenopathy. The 6-year-old is placed on 4-drug treatment. And the point is there's 2 things with the skin test. First of all, or the IGRA, if you place it early, after exposure, it can take 6 to 8 weeks for the immune system to learn how to respond and make a positive test. So if you actually test it early, you want to test early because you want to sort of treat people and evaluate them right away. But they could be negative just because their immune system hasn't been primed yet to actually respond. So in some countries, what they do is they like to repeat the test in about 8 weeks to see what the result is. But in this case, the other reason is that sometimes when people are sick or they have immune systems that are compromised, they actually won't be able to react to a test. And we know that with the interferon gamma release assays because they have an indeterminate status where if you can't mount to the control, we know that you wouldn't be able to mount a response even if TB was there. So we call that indeterminate. With the skin test, it's just going to read as negative. So the immune system also plays a part in it. And somebody, you can imagine if they have really bad TB, they might be skin test negative. That's just to really reiterate what Dr. Vrinder was saying before. Okay, so we've got 2 kids and a mom who all had TB on the contact screening. The other older 2 kids had normal chest x-rays, they had positive PPDs, and they were started on TPT. In the US, it's INH prophylaxis for 9 months. So one thing you can do is you can just look back on this, right, and see. So it's the younger 2 kids that had TB. And part of it is the younger kids are going to be staying home. They're going to have more exposure to, in this case, it's mom, right? Mom has TB. The older kids have other sort of program activities and they're going out more. So they might have had a lower load of exposure and also probably better immune systems. So all these things kind of come into play when, when sort of looking at and seeing whether somebody goes on to have TB infection that's controlled versus sort of disease, but it's fascinating. So I think that one of the things that's very important just among the TPT for children is it's an opportunity to actually discover TB disease earlier. Because if you wait just for symptoms, you would have waited for a much longer time, right? All of these folks were pretty asymptomatic. So you got earlier diagnosis and then also the TPT to prevent TB for the 2 older children. Okay, next slide, please. Okay, so I'm just going to highlight this. There's a lot of questions that I hear in sort of in areas where the concept of TB infection and TPT is still new, like how do you know it works, etc. So a meta-analysis and systematic review was published in 2020, and it used actually individual patient data. So that means that they go back to those studies, get the original data, and then redo the analysis. So they looked for studies that followed child contacts, so close contacts, and then those studies followed them out for TB incidence, whether they got TPT or they didn't. And they got 46 cohort studies from 34 countries, and this ended up in 1.3 lakh children that were followed for 4.3 lakh person years. And you can see that there were 1,299 prevalent cases and 990 incident TB cases diagnosed. So what they were able to see with that was that if you looked at all child contacts, whether they were positive or not for a TB test, it was 63% effective. So much lower TB in those that took TPT versus those that did not. And then if you looked at the contacts that only had a positive TB test, it was even higher. It was 91%. Okay, so let's just kind of review what we know about these states of TB infection and TB disease. This will be a review for some of you, but it might be also still new for others. So for people that have TB infection, the bacilli is dormant. It is it's not dead, it's still alive. I always say it's in a hibernating state. It's controlled by the body defense system, either walled off by granuloma or in other ways. If you get a skin or a blood test, it'll be positive. So the IGRA or the skin test. If you check for smears and cultures, and I would say gene expert, they're going to be negative, and we don't consider them to be a case of TB. They're not ill, they're not contagious, their chest x-rays are normal. Okay. And on the other hand, somebody with TB disease, the TB bacilli are actually actively replicating. They're not dormant. They disseminate. They can go anywhere, cause disease. The person feels sick. They're contagious. Usually we'll have a positive skin test or IGRA test, and they're considered active. Keep on going. All right, and so what I'm showing you right now is sort of the classic understanding of TB infection and disease. So here's the classic sort of pathogenesis of it. You have somebody in that upper left corner that has pulmonary TB. We estimate that they infect about 10 persons a year. And so among those that are exposed, about 50% of them will become infected, and some of them, and 50% won't. This really depends on a lot of different host, environmental, and contact factors, right? So for the host, it depends on how contagious are they? Are they cavitary? Do they have cavitary TB, laryngeal TB? Are they in a small enclosed space that doesn't have ventilation, meaning the dose of bacilli is going to be really high? And what about the host, right? Is the host immunosuppressed, immunocompromised? Are they very sort of healthy? All those things kind of play into this part of the primary infection, the dose. Then from there, what can happen is one of two things. So 5% of them will move straight into active disease, and that's primary TB. So primary TB disease, and that looks actually somewhat different than reactivation TB. So 5% go there, and then that's like pleural effusions or disseminated TB, miliary. 95% go into this kind of latent state. And from there, another 5% have a lifetime risk of developing TB over their lifetime. That's why we say there's about a 10% risk, right? And that can go into pulmonary or EPTB. Okay, next slide. All right. So that rationale for treatment of TB infection is that this really is the repository from which future TB cases can arise. And without sort of addressing this, you're not going to be able to turn off the tap of TB. So I would say, like, even in things with active case finding, right? So if you do active case finding, which you may have heard a lot about, they go out and find all the TB cases. That's great. You've found the TB cases for now. But if we don't address the reservoir of TB infection, all those people that are infected still have the potential to progress a TB, in the future. It is the highest for that first year or two after exposure, and then after that, it depends on their immune status. Okay, next slide. Okay, so it's interesting, our belief and structure of what we thought was TB infection and disease was very dichotomous before. It was kind of what I showed you. It's like one way or the other. And then, what happened was we started to have TNF blockers in the early 2000s, and you can kind of scroll through the next. I think I didn't know that I put it on the animation. So what happened is when these TNF, go back. So when the TNF blockers came on, go back. Yeah, there we go. Perfect. When the TNF blockers came on, what happened was the rheumatologists realized that they started diagnosing a lot of TB, and they started reporting it. And what we started realizing is that there was something with in the tumor necrosis factor cascade that's important for keeping TB in latency. The next slide, please. Yeah. So where we used to think that, you build that granuloma, it's like that calcified granuloma, it's like a house, and it just contains the TB bacilli. It's more like it's an active process. There's an active process that is required to maintain the granuloma. And when the immune system is missing something, like the TNF part of the cascade, it cannot retain that granuloma, and the TB is able to escape and basically replicate. So it really highlighted how the cytokine cascade is crucial for maintaining cellular defense against MTB. Okay, next slide. Yeah, and so this is kind of that same thing, that really maintaining latency is an active process. I know I hear a lot about it in India when we talk about nutrition as prevention, because we know that nutrition is also tied to being able to maintain healthy immune processes. Okay, next slide. Okay, this slide, I just wanted to show that some of the work that's actually in macaque monkeys, this is the model for, there's just a few models for animal models for latent TB. I think I heard Dr. Schreier talk a little bit about this. It's pretty hard. So guinea pigs are one, macaque primates are another one. And so they've actually been doing these studies where they do the functional MRI and they can watch the granulomas and they can see that they're in different stages of evolution. So it's not, even in the body, it's not a 0 or 1 state where, you know, everything is latent or everything is active. So you have different granulomas that shrink and grow, you know, so it's more like a, you know, the whole ecosystem. Okay, next slide. All right. So our understanding today is just more subtle around the spectrum of TB that can manifest. So we have, you know, the clinical disease, which we all know about with active replication and disease, and they're sick. But we also have subclinical TB where they might not be necessarily symptomatic, but there is metabolic activity of the bacilli. You might think about that mother who barely had symptoms and had a pretty normal chest x-ray, but actually cultured out TB. That would be, you know, sort of a good example for that. And then, you know, there's incipient TB where they're, you know, on the edge of kind of becoming metabolically active, but not quite. There is, you know, infection, like sort of deep dormancy. And there's also like, the innate immune response where people actually become infected, but they can eliminate it on their own without the antigen presenting T cells. Okay, next slide. Okay, so the tests that are available, basically there's the skin tests, PPD, and now CyTB as well, which has been approved in India. The interferon gamma release assays, the two most common ones are the quantiferin blood tests, the T-spot. There's a bunch of others that are actually available in other countries, but not available, I would say, globally. Okay, next slide. Okay, so just here's a simple schematic of how these tests work. They're all based on the same thing. So sort of the first step that happens is you have an antigen presenting cell that presents the TB antigen, and it sets off this whole cascade, right? So the memory T cell kind of bonds with it, you have the compound, and then you have these cascades, the TNF cascade, the interferon cascade, the interleukin cascade, you know, on and on and on. So in the skin, it's just that you have a type 4 delayed hypersensitivity response. That is what that wheel is. So that's what develops because you had to have seen it before, right? In vitro, it's just that you're only using those specific antigens and then you're measuring something. So with the interferon gamma release assay, you're measuring the levels of interferon gamma. You could, you know, maybe have like look at other factors, TNF factors or IL-8 factors, and they have been studies with that, but the interferon ones have been the ones that have been the most successful. Okay, next slide. Next slide. Okay, so just a little bit about CyTB. So the CyTB is the skin test, but the only difference is instead of using the PPD, they're using specific antigens, the ESET6 and the CFP10 antigens. And because of this, you don't get cross-reactivity with other mycobacterial species, so it's more specific. The pros about using something like CyTB is that it's low cost. You don't need advanced laboratory infrastructure for it. You can kind of, you know, have somebody to place it and then just read it in 2 days. The cons are that you have to have training for those folks to be able to place the skin test. Is not absolutely foolproof, and also the reading. They have to be trained for that too. 2 visits are needed. and the other kind of cold chain requirements. It is approved for use in India, and NTP is, I think, planning on using this as their primary test. They're just negotiating right now with Serum Institute. Okay, next slide. Okay, so this is just a quick slide. This is actually an industry slide of quantiferin when it was still owned by Celestis, and it shows the different cross-reactivities for the different mycobacterial strains. And you can see with 10 that it only really interacts with the MTB complex, and Cassasii, Marinum, and Zolgii. And most importantly, it doesn't cross-react with the BCG substrain or avium. Okay, yeah. And so you see, this is out of a large cohort study that was done in the US where we compared TST, quantiferon, and T-spot. And you can see the different age ranges. And the things that I would say is, the things to look at is, if you look at those that are TST positive and interferon gamma positive, so quantiferon T-spot positive, it's the red bars. And you can see that they really correlate with age, meaning that it's low when they're younger and it kind of goes up with age. And if we look at something that we want to have as a marker for TB infection, one thing that we know is that, it's going to be associated with time that you're alive. If you haven't been alive for a long time, your opportunity for exposure to TB is just a lot less. So, you might be a contact, but, just in general, time is going to play a factor. If you look at the blue bar, which is the TST positive and quanti or T-spot negative, so these are discordants, you'll see that it's the inverse. So the less than fives have a huge proportion, almost 90% are TST positive and IGRA negative, and it goes down over time. So for us, this is one of these like ecological studies that show that at least in the US, there's a component of TST positivity that's related to BCG. I see that in the US because I think that there have been some really good studies in India that showed TST waning positivity after about 2 years after their injection. So there have been a few studies that have been done here in India, but not, there's some, there's some issues, and one of the big issues is going to be you're in a high prevalence sort of area, so there's reinfection and things like that. But anyhow, okay, next slide. All right, so now let's go on to how we came up with the sort of the TPT, the treatment regimens. And there's a lot of studies out there. I've really just focused on the original studies. So we can just kind of go back to the original ones that showed us what we know and why. And then there have been many studies from many countries that have sort of replicated, et cetera. So this is a very early study that came out of WHO, and they looked at different lengths of time of taking INH. So, and then what the risk reduction was, what the compliance was, and what the risk reduction was if they were compliant. So one thing you see is with the shorter, like 3, 6, and 12 months. So the shorter the time is, the less effective it is at reducing the risk of TB, right? On the other hand, the shorter the time is, the greater compliance, right? It's really hard to stay in INH for 12 months. So they had 70% of them, but you can see that the effectiveness is 93%. So it's a really good regimen if they can stay on it for that long. About 70% for 6 months and only 30% for 3 months, which is why we never had a 3-month regimen for INH. Okay, next slide. This is one of the original actually randomized controlled trials ever, but it was looking at household contacts and IPT. Look at the date, it's 1962. They had 12,000 people in each arm, one for INH for a year, one a placebo, and they looked at progression to TB. And you can kind of see below there for both extrapulmonary TB, 4 that were in IPT, 16 on placebo. for extrapulmonary, and for pulmonary is 14 for INH and 64 for placebo. So really, relative risk of about 4, if you didn't take the IPT. And this is one of the sort of first trials that kind of planted that this actually works. Okay, next slide. Okay, so that was a regimen that was used for a long time. It wasn't used very much because it's a very long regimen. People don't like to take drugs every single day if they don't feel sick. And also if they can't drink alcohol. I mean, that's just like a reality for a lot of people where that they're not gonna take the meds if they can't, have their glass of wine in the evening. So there was, been a hunt for another regimen, and this is from 2011, the randomized clinical trial that proved the equivalence of 3-HP compared to 9H. So this is the prevent TB slide, and you can see that the difference in rates is very small. It actually favors 3-HP here down on the bottom with the blue, but it didn't quite reach significance. Okay, next slide. I'm kind of brazing through these because, I want some time for a discussion. All right. So again, this was about 4,000 people in each arm. And you can look and see that the treatment completion was hugely different, right? It's so much easier to finish 12 doses within 3 months rather than 9 months. So that's huge. And then you can see that developed TB was, not so different. Adverse events is like not so different. Hepatotoxicity, much more common in the 9H with isoniazid, and then hypersensitivity, much more common in 3HP. Keep on going. Okay, and then the latest, which is 1HP. So this came out in 2019, just pre-pandemic, and this is looking at 1 month of daily rifapentine and isoniazid compared to 9H for PLHIV living in areas of high TB prevalence. And you can see again, there's equivalence. Those lines are right on top of one another, and basically the TB incidence was 2% for both 1HP and 9H. The SAE, so serious adverse events, was also about the same in both arms. So 6% in 1HP and 7% in 9H. And again, the treatment completion was very different. I mean, these are really high rates of completion for 9H, 90%. So those people were very motivated. Usually it's kind of lower than that, but still almost 100% for 1HP, 1 month of daily rifapentine and isoniazid. Okay, next slide. Okay, so this is just a systematic review that goes over the efficacy and safety of the different regimens in TPT. I'm going to just keep on skipping. I'm going to skip this. You can look it up if you want to know more. But basically, it showed the things that I showed before. A shorter regimen leads to higher completion, more hypersensitivity in the 3HP, less hepatotoxicity, etc. So keep on going. Okay, so this is another trial that I'll just sort of highlight. This was done, Neil Martinson did this out of South Africa. There are 4 arms to LTBI. So it was 3HP and there was 3HR, 6 months of isoniazid and then continuous isoniazid. So they looked to see what the difference were. Actually, this is where continuous isoniazid came out of because they had the best sort of risk reduction coming out of a very high TB prevalence area. Next slide. Okay. However, the rates of hepatotoxicity were also the highest. So you can see that rates for 3HP were 1.5%, for the biweekly 2.4%, 6H 5.5%, and continuous was 28%. I bring this up because I think the rate of hepatotoxicity is really, it's really tied to the exposure to isoniazid. Yeah. Okay, next slide. Yeah, so that 1HP one was more in PLHIV. And I have to bring some of these up because, first of all, it's very tricky looking at TPT studies because you're going to see this, but the number of incidents is very low, and for kids, it's even lower. And the diagnostic sort of ability is low. But PLHIV is kind of, that's a good population to look at just because they are somewhat immunosuppressed. So if they have bacilli that have not been eradicated and are going to progress, they're going to be more willing to do that. So yeah. Okay. So here, I'm just showing you this this template form for anybody who's going to be interested in doing sort of research. This was used actually for a post-marketing surveillance project. It was actually used programmatically, and we were able to track ADRs and AEs easily by each dose and kind of follow up what happened. There's a lot of concern when 3HP First was the results were first published in the US because we'd had some disasters with another regimen. And again, the big thing is like these people are healthy, so we don't want to do anything that's going to be bad for them. Okay, next slide. Okay, so just quickly, this is in the US. This is that first post-marketing project. We had, a little over 3,000 patients. The main thing is that the really high completion rates, even in the program, 87%. They're about just a little over 7% with symptoms. And this gave a lot of confidence to the programs across the states for them to take up 3HP. Okay, next slide. Okay, so in India, we had a CDC, we had a few of the early demonstration projects to see how this would go in India. Because there's always, you always are worried, you're always about adverse events, if it's going to be affecting a population differently. Also uptake and acceptance. And so this is among household contacts in Nagpur. And you can see that we had a little over 1,000 household contacts. They're screened for TB. We were able to find 8 cases of TB. And then in terms of the TPT, we have 1,030, so 92% that were willing to get tested. And there are 45% of them became positive. And the, oops, what happened to this? 83% initiated. And I was going to say the completion rate just got cut off. Let me see if I can get it. Oh, it's just cut off from here. But the completion rate is around 90%. It was quite high. Okay, go on to the next slide. This is looking at the ADRs. This is something that's very important. I've had people from outside of India ask sort of how this has been in our population here, and it's been pretty good. So about a little over 1% of the household contacts discontinued because of ADRs, and the number has been low. The ADRs have been mainly nausea and vomiting. I'll just say a word about this. We found this in the post-marketing project as well. Nausea vomiting is the most common symptom. It does not necessarily lead to discontinuation, especially if you can pre-treat the symptoms. And the symptoms tend to come on, this isn't published yet, but it's most commonly experienced and reported around dose 3 or 4. So those kind of early doses are the ones that are important to monitor for. Their abdominal pain and 1 instance of fever and chills. Okay, next. All right, this is another project looking at LTBA prevalence among household contacts in Mumbai. We had about 500 household contacts that were enrolled. And interestingly, for here and the other one, there are about 75% of the index cases had at least 1 household contact with TB infection. And there was about 25% that had every single person infected. We actually found that at both sites. Yeah. So the prevalence here was higher. This is in Mumbai. It was 51%. And then they saw here that persons that were younger, the children, had lower prevalence than the other ages. I think this makes sense even if they're contacts. I mean, they've been alive not as long, and they have so everyone has exposure in the household contact, but the people that are older have also had opportunity to be exposed in other settings as well. Okay, so just completion rates here in Mumbai, 84% initiated treatment and 90% completed TPT, 11% discontinued. And you can see that it was like about 3% due to ADRs, headache, fatigue, rash, vomiting, and 2 actually went on to develop DSTB. We always have to figure out with that, we haven't done the analysis for this yet, but whether it was a prevalent case or not, or it's reinfection from a different, source outside. Okay, next. All right, so now I want to just, yes. So I'm going to just quickly go through this quick, sorry. Or we can, I mean, I don't know how to do this because I knew this is going to be too long. So, I mean, maybe I'll just quickly kind of breeze through this. So I wanted to bring up like Chai and Find and Gates because they're doing these G 2.0 projects, huge projects with TPT. Right. And so I think there's definitely learning to be had from them. So this is from Chai. I'm not going to go through this. So just go to the next slide. These are some take-home points. So I think the take-home points here are that the acceptance of TPT initiation depends on when you actually go to talk to them. So the earlier you go, the less delay there is in the assignment, the more willing they are to take the TPT, and it goes down with time. We found this in the US too. Okay, then the other one. Next slide. Okay, and here they like improved and figured all this stuff out operationally on how to get the chest x-rays ramped up, but there was a drug shortage. So you have to have everything working together. That's the other piece, like everything has to work together. You can be doing everything perfectly, but there's other pieces that are just completely key. Next slide. Find. So Find also had a large project. They had specific pilots that actually did IGRA testing to initiate TPT. I think one big thing is that without testing, it's very hard to have acceptance for TPT, because people are going to question why they need to take it. Next slide. Okay. So just some key learnings. I mean, this is very programmatic, but engaging frontline workers, shorter regimens are definitely better, decentralization, coordination with the whole of NTEP as well as private providers. Okay, next slide. Okay, so this is, I hear a lot about people worrying about the risk of development of drug resistance through TPT. There's very little data on it. There's basically 3 studies, and I have them here. One thing, remember, like the numbers have to be huge to be able to show a difference. You've seen this in all these studies. That's why they're expensive and sort of hard to do. But this one is from 2006. It's still like the major systematic review that has been done. And you can see all of these different studies that have been, have looked at this. And the main thing is the estimates there's some that fall on the side of development of INH, isoniazid resistant TB, and then those that fall on the other side. But the confidence intervals are so wide, they all overlap 1. So it's like, that's why they say like, you really can't say anything. There's no evidence that really strongly suggests that, giving TPT, puts you at risk for developing isoniazid resistant TB. And they used a few different definitions of resistance. Okay, next slide. Again, this is not children. This is PLHIV in a very high risk setting, South African gold miners, for the reasons that I kind of told before. Very few studies in only children, but you can see basically this is programs where they did a lot of really active, IPT and case finding, and then compared to the control clusters and labs. And again, there's really no difference. In fact, sometimes it looks the other way. Okay, next slide, last slide. Next to last slide. And this is looking at, go back up, development of rifampin resistant TB. So there are, they're kind of in 3 different stages. The main thing is look at how low the numbers are. They're really few. And most of these were in PLHIV. So there are 2 isoniazid resistant TB TB cases when they were given arifamycin that was in the BMRC studies, a few PLHIV. Next slide. Yeah, so the Prevent TB study, there was 1 person who was HIV positive, actually had diarrhea, so there were absorption issues, late treatment completion, and in the Martinson, PLHIV as well. So I think the overall message is that the levels of resistance are really, really low. They're really low. That's why it's hard to say anything. The confidence intervals are going to be wide, but there's nothing that actually supports our kind of worry and concern about development of drug-resistant TB. Okay, next. All right. So just a few thoughts on research gaps and opportunities and ways forward. We talked about the spectrum of TB today, and I think that really the vast majority that have TB infection are not going to be progressing to TB. So if there's any way to detect those that are most likely to progress to TB, then we'll be able to focus on those that will most benefit from TPT. So I think a lot of work with incipient TB, diagnosing them will be helpful. There's been a lot of biomarker work already through SATV and Report Consortium and others, and I think I heard a little bit from Dr. Ashwarya. It came out of the vaccine studies, actually. Other modalities of testing, cell-free DNA, targeted NGS, just different. Is there a way to be able to tell drug sensitive from DRTB, LTBI? That's not even on the books yet. Vaccines, prevention, you heard all about that before. Treatment, I heard a talk from Jacques Rousset, and he's, we can think big. Is there a way to get treatment down to 1 to 2 weeks? So much easier to do. Long-acting compounds, depot shots, single shots. Definitely we need TPT regimens for MDR and pre-XDR TB. There's quite a bit of fluoroquinolone resistant TB that's around. Is there a biomarker for TPT success? So Dr. Rinder talked about this in his first, his first talk. It would be great to be able to show somebody that they've been able to get rid of their sort of bacterial load in there. And we don't have that right now. And then programmatically, interventions that increase acceptance and uptake of TPT by both health providers and community members. So that's it that I have, and sorry that I have gone over. You can flash the acknowledgement slide. There's a lot of people to acknowledge, a lot of programs that have done work and contributed to this. So that is it. And I think I saw a lot of questions sort of flash by. So let me see. Okay. So what is the approximate prevalence of LTBI in Indian children and adults, respectively? So, that is just from doing the TBEST study with the 21,000 people. The question needs to be divided up. It can't be answered just like that, because for one thing, people that are contacts, known contacts, so household contacts or other close contacts, their prevalence is going to be very different than the general population, right? Because they're a known contact. So that's one thing. And the other thing is that we actually don't have, we'd had some estimations previously, and they've been a little bit all over the place. I would say there's some 30%, some 50, I think some 70%. I think Dr. Rinder will know some of this. During the National Prevalence Survey, there was an effort to piggyback a latent TB prevalence survey on top of it. So we could look at that, and that was done with quantiferin, and it was done in different cities. The way that it was designed, and this was mainly because of, I think, just, it has to be so huge if we're really going to do a true LTI prevalence survey. They couldn't do that. But so they did sort of localize prevalence for each of those cities. And again, there's some variation, 30%, 50%. Delhi is actually 70% the prevalence for LTBI. This is from, and I think from what I had heard from Dr. Karen Roddy, it's not, it was not done in a slum. So I think it varies, and I don't think we have an overall national prevalence, but I'll actually open it up in case somebody else knows.
Yeah, Christine, yeah, that was right. The total national prevalence came, that was done among adults above 15 years, not, we don't have data for less than 15 years, but
Yeah.
that came to 31.2% national prevalence range.
Yeah, but I was told that we actually can't sort of group them all together, that we're not supposed to do that because the way that
correct.
Yeah, the way that they're, but so the range is sort of, sort of below and then all the way up to 70% or 73%, something like that. Yeah. Okay. So the next, so the next one, 6 to 7% SAE and 1 HP are these are in all age groups. There's very few pediatrics in there. Actually, it's mainly PLHIV. So again, I'm sorry that these studies don't have that much information on pediatrics. Although what I can say is for the 3 HP in the post-marketing project, that both the adverse events and the severe, there were no severe adverse events in the children, and there was lower adverse events in the children in general, and that's actually published. Okay, are there any studies in LTI prevalence in vulnerable populations? Yes, there are. I think, in India or, I mean, so like PLHIV, there's a lot, and it goes by like many different countries. So one thing I want to say, and this also will relate to, there's a question I used to hear a lot, I don't hear as much, which is maybe good, is how long does TPT last? How many years does it last? And this is kind of the same question of LTI prevalence. What is the LTI prevalence? The LTI prevalence is completely related to the TB prevalence. Because remember, the people are getting TB infection from TB cases. So the higher your TB prevalence is, the higher your LTBI prevalence is. And so there should be a relationship, right? That's the LTBI prevalence is actually an indicator of how much TB you have circulating around. If you have a really high LTBI prevalence, that means that there's a lot of TB around. And if your TB notifications are low, you should be worried that you're actually missing TB. So that's how I'm going to answer that one. And in terms of how long TPT will last, it's the same answer. So what TB preventive treatment does, it's not a vaccine. What it does is actually killing the TB bacilli that are dormant inside your body. They're intermittently metabolically active. And it's during that time that they're intermittently metabolically active that there is bactericidal activity with the antibiotic. It's a lower load, which is why it works. So once you finish your TPT, you've reduced your LTBI bacilli burden in your body so that the chances of being able to break out into an active disease state is much lower. However, if you get exposed to TB again, we say exogenously from somebody else, you will actually be at risk for getting TB again. And if we think about it, right, we know that people that have TB once are not unfortunately protected from getting TB again. That doesn't actually give them protection. This is, I think, the big challenge with the vaccine, is trying to figure out what's going to give you protection? Because even when the TB doesn't give you protection, you know, sort of what is going to do it? And it might be sort of what Dr. Avrinder was saying, might be some other host response that's going to be sort of the winning bullet for that. You know, because we do see people that actually clear the infection without the T antigen presenting cell. So, okay. I don't see any more questions.
Yeah,
So-
Christine. Yeah, Swati.
Yeah, so I have a question. Is it while using an LTBI testing, probably the upcoming test, skin test, which is coming up, is it prudent to always use it among a risk population than going into a general population? Always be
Okay.
risk mapping done before we apply the LTI testing in a heavy burden setting.
Yeah. So I think what I'm understanding your question is it's about focusing resources. So it's like, who are you going to be doing this TB test in, right? So, and I would say it is smart to focus your resources, but you know, so you want to focus them. I would say don't focus them in a checklist type of way, which means that sometimes we have this checklist. Oh, you know, diabetics, blah, blah, blah. You need to think about it in a disease epidemiological way. So one thing that I would always say is like, so homeless people that don't live in houses in the US, you know, they're a population that is at risk for TB. And people would always ask me about that. And we did these studies in the TB Epi Studies Consortium. What we found was if there's TB circulating in that community, then you have a problem with TB in the homeless. If there's no TB in that community, you will not find any TB among the homeless there because it will not auto-generate by itself. It has to come from somebody else. And so I think in that same way, you want to, when you look at the risk population, you can look at the vulnerabilities that we usually think about, but also look at other things that tell you that there's a lot of TB going on. Are there TB deaths? Are there people that are diagnosed that are disseminated TB or very severe TB? Those are sort of markers of delayed diagnosis. So yes, that answer is yes.
Thank you.
Yeah, Swati, again, I hope Dr. Christine has answered those questions. Again, there are two ways we can look at that. There are some of the countries which moved into community-based TPTs, but we still focus on risk group-based TPT approaches. So, I mean, even with the risk group-based approach, there is a huge policy practice gap that is actually we are looking at. We are thinking of India, the population is so huge. If you look at in the last year official report itself says only 58% of the eligible children under less than 5 years were put on TPT in India. 58%.
Yes.
And if you look at more than 5 years, no, out of eligible, maybe hardly 15%, maybe 15% might be put on TPT. So there is a huge policy practice gap still exists. We also, I mean, so as if you look at from the epidemiology, logistics, operational point of view, I mean, we can go ahead with focusing on vulnerable population first, as far as TPT is concerned, and go about that. But the thinking is basically we need to scale that up quickly. We need to scale that up of TPT. This is my personal opinion. I mean, there may be different opinions, but at least in India, let us focus on the vulnerable group first and scale that it up to the best possible extent. Because there is other challenges also. So for example, ruling out active TB is something which is the crucial step when we are going ahead with a TPT. So we need to rule out the active tuberculosis. This is a very crucial step which we need to, which is the basic bottleneck that we are facing as far as TPT is concerned. These are my thought process. Maybe Varinder sir might have some other thought process.
No, I agree with you, Dr. Rakesh. The only thing I would like to add is that there is a third player in this, and that's the patient himself, who's the most important player. So many times the acceptance for a putative benefit is not taken by them. So I recall one of the studies which we were looking at about 5 years back on prevalence of TB amongst the household. Despite providing them free X-ray, free access, you walk in, we'll do it, no queuing, nothing. They said, no, we are healthy, we don't want.
Yeah.
So the willingness to walk to the hospital to get it done, loss of wages, there's several more issues. And I think that the bigger issue is also about, here it is an active drug which you are taking. You are always scared. Will it harm me in some way or another? So the benefit is putative. I am healthy. Yes, somebody in the family had TB. I'm okay. I don't have anything. So the fact that I don't have any symptom, it makes it less acceptable, because once you are suffering from symptoms, then there is a motivation to take care of them. When you don't have any symptoms, that more important motivation goes down. You can counsel, you can explain. I would not say not. I mean, I have found it easier for families to accept preventive therapy for children than for adults. They accept it for children. Okay, maybe it's a small child, it is a higher risk, even if they're asymptomatic. So not a large experience, but whatever where we have talked to the whole family, it's often that, okay, I'm okay for my child.
I agree with you, Dr. Rinder. That's why I actually put it in one of the researches sort of topics, is increasing acceptance for it, because I think it's huge. You know, in our projects, we had high acceptance, but there was a lot of education, and the education went not only just to the patients, but also to the private providers as well. You know, and so there has to be sort of an understanding of it. So I think that is huge. And the thing that, you know, I said, that's first do no harm, right? So the first thing is first do no harm. In the US, we had another regimen. It was 2 months of rifampin and PZA. I don't know if you know about that experience, but that got touted, and that was through, yeah, that was mainly shown to be effective in PLHIV. And then, you know, CDC had a recommendation, and people started using it, and there were like cases of fulminant hepatitis. It was huge. And so with 3HP, that's how this post-marketing project came to be. We had to be so careful and so on it, because any slightest thing is just gonna, you know, you don't need negative sort of publicity. You also don't want to be doing harm. It has to be helping. They're healthy. You know, it's to prevent a future risk. Yeah.
So those are the challenges. I think the challenges are safety, and then it's a whole big question about counseling, and another question about suitability, eligibility, whether you are ruled out disease, whether you have the wherewithal. And then one of the slides you beautifully showed, when everything was available but drugs were not there, it went to not. So there are too many things to be fixed to get it going. But then I think the initial steps are to talk about it, to create awareness that there is a need amongst the professionals, so that it then percolates down to the patients and their families, and then have the policy to make it available. So it's a gradual step. Yeah, we are running out of time, so we need to be fast against this disease, just as we are running out of time for this seminar today. But I think everything needs to be on board. So thank you, Christine, for the brilliant talk on preventive therapy.
So there is one more question.
Sure.
Yeah, there's one more question. Is it sufficient to do a chest X-ray and TST in children less than 5 years who are exposed to TB? Is there any role of expert or smear at that stage?
Oh, you mean without the
it's talking about household investigation. Yeah, it's talking about investigation.
Okay.
Contact
You mean to do an expert on everyone? I think the biggest challenge is, how are you going to get the samples, right? It's so hard to get samples in children that are symptomatic. Forget about children that are not symptomatic. I mean, that's one of the things, right? Even for adults, that sort of end of treatment culture or smear or whatever that they needed, they're not coughing anymore. How are you going to get that sputum? So I think that that's hard. I mean, maybe one day we'll have something else that's not a sputum. That's, I don't know. I mean, with stool is kind of still challenging, but
you can't
yeah,
tell what does not exist. So the patient does not have
exactly.
respiratory secretion. How do you check it? So it doesn't make
Exactly.
sense. So
Yeah.
the only thing could be that, okay, sometimes the cough guts aren't reported or they may be very mild. So if you see something on X-ray, then okay, possibility of disease, you go
Right.
a different pathway. But in children, what's recommended is largely a symptomatic screen. However, that's the national policy. In our setting where it's easy access to investigation, we do go for PPT when it was available. Now we do go for an X-ray. But at a public as a public health strategy, that should not become a hindrance for getting a TPT, because these young children are at a higher risk of exposure to infection. So and then reading of X-rays, it has its own difficulties. So given that scenario, if there are no symptoms and that is used to rule out disease, because that's a pretty sensitive and less specific way of looking at it. So since it has a fairly good sensitivity, there are very few patients who would have an active disease and will be totally asymptomatic, which you will miss. I'm not saying, unfortunately, medicine is never in a 2x2 table. So you always have some who would defy any rule or any box. But then you look at the larger picture. In the larger picture, the symptomatic screen works. But yes, if facilities are available, you can do an X-ray. Go ahead, do it. As long as it
Yeah,
gives.
and I'll add one thing to that. So I'll say that say even they had TB, like we had these kids that didn't have symptoms, right? They had TB. Say you did the symptom screen, you didn't pick up the TB, and you put them on the TPT. Most of the time, it's still going to be okay anyhow. So we had a huge pediatric cohort in the US earlier on, where there are a lot of children that actually we found out that they had TB afterwards. But because it's a possibacillary disease, it ends up kind of being okay. They're either going to declare themselves. So they start having TB symptoms as their load kind of comes down, their immune system gets better, and then you diagnose it, or it's possibacillary, it's still going to be okay. So, I mean, I know that that's such a worry, but I also agree with you, Dr. Vrinder. It's like, it's better just to do the TPT. They're probably going to be okay.
Okay, so are there any more questions? Okay.
Shruti has the question.
Yeah,
Yeah,
I
so
just wanted. Hi, this is Shruti Polishwala from Serum Institute. Thank you,
Oh,
Dr.
hi.
Ho, for the presentation, and I saw that you'd included SciTB in your slide deck. Maybe not today, but if you have the time, we can definitely go through the SciTB presentation, because when you were speaking about the under 18 children, or as I said, SciTB can be used for testing of latent TB for anybody over the age of 28 days. So we can get into the details of that. Maybe on another day that you are free, just to show you the data on the clinical trials that have been done. Of course, as you mentioned, we are in talks with the government, and the data of the under 18 should also be coming out soon. So I think that could be a good option for exploring the testing for the below 18 as well. Thank you.
Have you submitted the data for DCGI approval for users below 18?
Yes, sir. That's currently under evaluation. So what we will do is, whenever, maybe right now is not the most appropriate time to present, but we can organize a session wherein we can present to you just specific for SciTB, of course, so that at least the system is aware of the product and its advantages and how it can be used.
Oh, definitely. I mean, the people are in India quite used to using skin tests. So, despite its challenges, and this is a better product, being more specific. So, this is something which the profession has been looking forward to. But then, as you know, we are all kind of constrained by the regulatory approvals. So, we look forward to learning more about the product once it is approved for children below.
Absolutely. We can organize something.
Right. Sure.
Thank you.
That would be great. Thank you.
So
I think so. There are no more questions. We can, Swati?
We can summarize, Chair.
So the quick points I would say to take home today is that we have some new technology which is already adapted. It needs to be made more efficient, closer to the place of working, whether it is to decrease the turnaround time or to make it more sensitive to pick up specimen or make it more amenable to different type of specimens, be it sputum, be it stool or be it oral swab. That's something where a lot more work needs to be done. How we can increase the repertoire of specimens which can be used for the range of specimen which can be used for testing by these tests. And the second, I think the important thing is the wish list which has come in, which talks of thinking differently. It's not thinking aligned only to a singular diagnosis, about a more robust diagnosis for exposed, infected disease, and linking it with a possible treatment. So uncharted area remains the host response and the cytokines and things like that. The larger issue being, can this be put into a cartridge like just like a CB0? Can it be made cheap enough, and can it be made in a machine or in a way that it is doable at the periphery and without much involvement of a technical person? So that's something which is on the wish list. There are products in between which are there. So that's what we heard on diagnostics. The second part was on vaccination and prevention. And we did learn that there is a large project going on revaccination, whether the newer vaccines, revaccination by them would be safer, more efficacious vis-a-vis the traditional BCG is something we need to look at, while we also find the answers to whether it is a good strategy to revaccinate in a high burden country like ours, where there is automatically some degree of exposure happening to people who are once they're vaccinated. So whether a further revaccination is going to be of additional benefit in a high burden country, something we really need to look at. With regards to preventive therapy, well, this is something which is more established. We have to find more acceptance within the profession and with the patient or within those who are infected. So to start with, we have to have a easy way of identifying those infected. Like I said, for children, exposure is a quasi parameter we use for they being infected, but that's going to be harder for adults. The adults, because they also tend to have more side effects to the preventive therapy compared to children who tolerate it better. Therefore, one has to look at these issues more diligently, whether they can be tested and treated or just treated without testing, or symptom screen would be enough or not. That's a kind of an implementational research which is important to do to answer this question quickly, and that will be useful in building into the national strategy for TB prevention. So once we have rifampicin available or a 12-dose regimen, it does take care of a lot of things around except adherence. What remains to be seen is, we saw if the INH was more on hepatitis, the drug's hypersensitivity was reported higher with rifampicin, almost a similar proportion, rifampicin group. So we need to look at that also very diligently, though the numbers are small. But then if the side effect is serious and you are giving for into an asymptomatic situation, then the acceptability for those side effects is much lesser. And that's something which is very key to take it further or to get it accepted across the board by the clinicians as well as by the patient. So these are my summary thoughts. Over to you, Rakesh, for your bit.
Thank you, sir. Once again, it's an honor to be co-chairing with you and the entire team and speaking to the young researchers of India. I take from what you mentioned, like we can all aspire for the moon now and even beyond, because India is the national superpower in space. With 40% of the country's total population being children and adolescents, is one of the world's largest population. And TB still remains the central of infectious diseases, causing huge number of deaths. It's alleged to be due to the absence of collective commitments for saving the young lives. We cannot continue to stand on these sidelines especially when our children are falling ill, especially when we know that we can prevent them from falling ill and we know how to treat them to do them. Christian was pointing out that the number of pediatric TB, it also means that it's an index of transmission also. It means that when there are pediatric TB in a place, that means there are lots of adult cases around. So we need to view it from the epidemiological point of view as an indicator of recent transmissions in the community also. We also need to look at the Constitution of India, which says right to all children. They also have the right to education. They all have the right to nutrition. They all have the right for standard of living and improved public health. We also need to advocate for making the right to breathe air free of mycobacterium and tuberculosis for all our children. And investment is really crucial when we move in that particular direction. For researchers, Sir has already mentioned most of the points for the researchers, especially basic science research. There are some few public health research questions or some social science questions which we can put forth is also. Maybe there are many fancy words, family-centric, integrated. So all these are very fancy words which we use theoretically, but are there good models of decentralized TB care services? Especially when Ayushman Bharat Health and Wellness Centers are coming up. How could we integrate pediatric TB services in Ayushman Bharat Health and Wellness Centers? How could we make it more family-centric? How could we integrate with other existing programs, for example, RMNCHA program? There are lots of things happening with RBSK, Rashtriya Bal Swasthya Karyakram, or RBSK or RKSK, even nutritional rehabilitation centers, even Anganwadis. So how could we integrate it further? How could we strengthen it further? All these are some of the questions which we can actually think of. Also, how can we empower communities, especially how can we make the voices of the children heard? And how could we strengthen some social accountability systems? These are all the questions which is quickly coming to my mind when we are thinking about pediatric tuberculosis. Also, we know that India as a country, one size shoe doesn't fit all for all context. So we may need to contextualize some of the local solutions also. We may need to bring out local solutions also based on some of the guidelines. Some of the places can move ahead. Some of the places need to follow. It need to be contextualized for India. So researchers can play a role in contextualizing or bringing about local solutions also. Sir mentioned about uptake for vaccines, uptake for TPT. All these things as challenges. We need to focus on that. We also need to strengthen the community health systems, like how community could be empowered to deal with for TB elimination. Like when we dream about a world where all our children are born in a world where they are breathing air free of mycobacterium tuberculosis, how could we ensure that community are leading that fight? How could we convert that into a people's fight against tuberculosis rather than a few people fighting it against tuberculosis? These are all the challenges. These are all the questions that is quickly coming to my mind from other point of view, from the public health and social accountability point of view. Thank you so much. I really enjoyed. Thank you, Sir Christine, and thank you, KEM HRC team for giving this wonderful opportunity.
Thank you, Dr. Varinder Singh and Dr. Rakesh for that chairing and wonderful sessions by the speakers. May I request all panelists to please switch on their cameras so that we could take a photo.
Participants.
The panel IDs, as in the participants, to please switch on the cameras so that we can take one photo. Also reminding that we'll continue our meeting tomorrow, same time, 2 p.m. to 6 p.m. So please do join. So let's, I hope everybody has their cameras on. Yeah, I think we can take.
You're ready.
Yes, ready. Done?
Yeah. Thank you.
Saved. Done. Done. Thank you. Thank you, everyone.
Thank you. Thank you for giving.
We'll join tomorrow at 2 p.m.
Thank you.
Thank you. Bye.
Yeah, bye, bye.